Good morning, everyone. Thank you for coming to our live session. We will have our first panel open up our day today, but with that, I want to actually turn it over to our committee member, Zara, who is online and will be moderating today's session. So Zara, take it away.
Muted.
Zara, you're muted.
Thank you. Yeah, I needed the ability to unmute. Thank you for granting me that. Good morning and welcome, everyone. I'm very excited today for this kickoff session of day 2 of this really exciting and invigorating workshop. So today we're going to start off with a session called Industry Perspectives on Environmental Safety and Security Considerations for Engineering Biology. This panel is relevant for many reasons, one of which being that the U.S. bioeconomy, currently valued at more than $950 billion and counting, and for accounting for more than 5% of the GDP, I think is obviously a rife for growth, right? This is a very impactful sector and will have increasing impact on the country's economic vitality. So my name is Zara Summers. I am the Chief Science Officer for LanzaTech, a gas fermentation company that's scaling around the world, hopefully including in the US. And I am a member of the National Academy's Committee on Environmental Safety, Security, Evaluation on Engineering Biology. So I want to thank the panelists who have volunteered to present today. And thanks for those of you turning in to watch this session. So today we're going to be hearing from Steve Evans, a Senior Technical Fellow at BioMade, Lauren Junker, Innovation Scout and Partnership Manager with BASF, James Duggins, the Head of Biosecurity for Twist Biosciences. So each of our panelists will start with 5 minutes of remarks, and then we'll do Q&A at the end. So now I'd like to turn it over to our first panelist, Steve Evans, and I will be timekeeping for you, Steve, so you have 5 minutes for your remarks. Thank you.
Okay, microphone on. Perfect.
Yes.
All right. So I'd like to thank you for the opportunity to speak to this committee. BioMade is a manufacturing innovation institute that was established in 2020 by the U.S. Department of Defense. We currently have over 300 member organizations, and our focus is domestic biomanufacturing. BioMade catalyzes the transition of proof of concept technologies across the Valley of Death to enable pre-commercial manufacturing. This corresponds to manufacturing readiness levels 4 to 7, which lie beyond the typical academic work at readiness levels 1 to 3. Lastly, our focus is on bioindustrial applications as opposed to biomedical applications. BioMade was established to solve problems in an industry-led manner. This extends beyond technical issues to include topics relevant to this committee. We frame these areas by promulgating a set of principles we called 4S, which includes safety and security. Relative to this committee's charge, our definition of safety encompasses environmental issues. Historically, these topics revolve around research in human genomics encapsulated by the ELSI framework. Our challenge was to coalesce these early research or technology topics into actionable outcomes of interest to industry that use biotechnology outside the realm of biomedicine to make the materials and products we use daily. The customary framings of safety, security, and environmental issues reflect an early-stage academic focus, and they garner significant interest in sectors of that community. But these discussions retain a low readiness level and consequently have little traction in later stage development and commercialization. Much like technologies failing to cross the Valley of Death, many safety and security and environmental issues fail to cross a chasm of relevance. This gap is sometimes discussed within the framework of the Collingridge dilemma, where Phil Shapira at the University of Manchester articulated this dilemma succinctly. In early stages of technology development, there is a dearth of data available to guide decision-making, whereas in later stages, technological pathways can become entrenched, rendering flexibility difficult. BioMade published the 4S principles and subsequently funded a project to evaluate how well these principles are incorporated in organizations contributing to the U.S. bioeconomy, and these publications were made available to the committee. Regarding definitions, we take an expansive view of these concepts. Safety includes steps taken to protect people and the environment from harm arising from biomanufacturing, development, processes, their products, or byproducts. Security encompasses efforts to manage potential threats or loss due to theft, misuse, diversion, unauthorized possession of property, including intellectual property, or intentional release of a biological risk. Interviews were conducted with 31 leaders across academia and industry to understand the perceptions of 4S principles and to hear barriers and recommendations. The top-level finding was that there was homogeneity in understanding safety, sustainability, and social responsibility, but there was heterogeneity in understanding security. The most common topics discussed for security were intellectual property, economic security, supply chain resilience, export control, and cyber or physical security. Only a few expressed concerns about biosecurity or harmful interventions by AI in biotechnology. This, I think, illustrates one dimension of the chasm of relevance, which captures the lack of translation of security issues in academic framing to practitioners of biotechnology at scale. Industry leaders highlighted the need to promulgate standardized methods to assess safety in its broadest sense to enable adoption and demonstrate compliance to best practices. Some tied safety barriers to complex regulatory pathways and felt that regulatory clarity would benefit biotechnology safety. Security saw the broadest list of barriers, including the extension of biosafety concerns about weaponization of fundamental research to all of biotechnology. More broadly, though, security barriers orbited around loss of intellectual property, physical or cyber vulnerabilities, and gaps in understanding regulations around export control or arms trafficking like ITAR. Broadly speaking, the leaders felt there remains a window of opportunity for the U.S. to promulgate impactful norms in industrial biotechnology as it expands globally. In conclusion, safety, environmental issues, and security remain important topics for our industrial biomanufacturing community, though the areas of importance to industry may differ from areas which may be framed in the communities focused on fundamental research. Overcoming this chasm of relevance is an important opportunity for the committee. Thank you.
Thank you, Steve. Thank you for keeping on time. Appreciate it. So we'll go over to Lauren for her opening remarks.
All right. Can you hear me okay?
Yes, you sound great.
Great. Okay. Well, thank you for the opportunity to present a brief introduction about BSF's strategic implementation of biotechnology and also our approach to safety, environmental considerations, as well as regulations. So, brief introduction, BSF is the world's largest chemical company with over 100 sites in the United States. And recently, we've reaffirmed our goal to be net zero by 2050. So, and of course, biotechnology is one of the pillars that we use to drive our innovation and sustainability strategy. Therefore, we also believe that the advancements we make in biotechnology can address some of the world's most pressing challenges facing our world today, including food security, sustainable chemical production, supply chain resilience, and also environmental protection. One of the sectors that we play in is the agriculture sector. We harness biotechnology to create crop protection solutions that enhance resilience against disease, pests, and climate change. And then through advanced plant breeding techniques, including genetic engineering and molecular marker identification, we do identify our farmers to increase yields while minimizing their environmental footprint. Our innovations aim to optimize resource usage, such as water, fertilizer, also chemicals, thereby supporting growing global populations while also respecting the environment. Looking beyond agriculture, BSF is at the forefront of producing sustainable chemicals through biotechnology. By using bio-based feedstocks, we aim to reduce our reliance on fossil fuels. We also use microorganisms and enzymes in production processes that allow us to create these materials that are more efficient and sustainable, with some examples including vitamin B2, acrylamide, an insecticide called Inscalis, aroma ingredients, and also industrial enzymes. As we explore novel frontiers in biotechnology, including synthetic biology, bioinformatics, and of course the use of AI, we recognize responsibilities that accompanies these advancements. Therefore, BSF prioritizes research in environmental safety by implementing stringent biosafety measures in our laboratory settings and also conducting thorough environmental risk assessments and gaining approval prior to any potential releases. This approach ensures that we mitigate risks to human health as well as the ecosystems in the environment. I know we wanted to talk about mirror life. So with this, as well as other technologies, we will most likely only engage in such technologies when there's a clear path for us to commercialization, and only gain that insight through consultation with governmental regulatory authorities, as well as others. For us, it's important to weigh the economic, competitive, environmental, and regulatory risk versus, of course, the economic, societal, environmental benefits very carefully. We do take a cautious approach to the development of new technologies, ensuring sound scientific and ethical considerations that guide our research and innovation efforts. So BSF does advocate for a balanced science-based approach to biotechnology regulation that emphasizes a clear, transparent, and predictable framework to facilitate innovation while ensuring safety and sustainability. We do support proportional risk assessment, harmonization of international regulations, and public engagement to build trust. Our position highlights the need for adaptability and regulatory frameworks to keep pace with scientific advancement, thus fostering an environment that encourages innovation while focusing on sustainability and addressing global challenges such as climate change and, of course, food security. So in conclusion, BSF's committed to leveraging our biotechnology to create sustainable solutions that benefit society and the environment, while also prioritizing safety, security, and ecological integrity. Through our initiatives in agriculture, materials, and nutrition, we not only enhance quality of life, but also are paving the way for a more sustainable future. So thanks for your attention, and look forward to discussing more today. So thank you.
Thank you, Lauren. So we'll turn it over to James.
Thanks, Zara. I appreciate the opportunity to talk to you all this morning. So my name is James Higgins. I represent Twist Bioscience. Twist is a manufacturer of synthetic nucleic acids based on the West Coast of the US. We have facilities in San Francisco and just outside Portland, Oregon. We're about 1,000 people. In terms of security, I think we see two major aspects that are relevant to our industry. The first, around biosecurity, DNA is a dual-use material, right? So you can do all the sorts of amazing things that folks do in emerging biotech, but there's a risk of misuse. So that synthetic DNA might be used to cause harm. And so that places a responsibility on companies like Twist that have technology that can make synthetic DNA to ensure that we're delivering that material to a customer who's going to use that in a way that's responsible. So since 2009 or 2008, the industry has worked pretty closely with the US government to both develop and then adhere to guidance around how we should think about ensuring that these orders won't be misused. Those recommendations from the government were originally published in a 2010 guidance document that really called out kind of 4 pillars. One, screen your customer, know who you're sending something to. The second, screen the sequence, know what it is that you're making and sending to the customer. The third would be to, if you have concerns about an order, reach out to law enforcement. And then 4 would be record keeping. Make sure you maintain records of all of the sequences you produced and who you sent them to, so that if there were concerns, there'd be a capacity to go back forensically and see what went to whom. So the industry has worked for a long time with the government to make sure that the major players adhere to these recommendations. In 2009, there was an organization formed called the International Gene Synthesis Consortium. I currently chair that organization that is comprised of 40 companies around the world, all working to both adhere to and improve these kinds of practices to make sure that we reduce the risk of misuse for synthetic nucleic acids. That guidance was updated in 2023. There was also a more formal framework that was published by the Office of Science and Technology Policy in 2024. All of these practices really are focused on kind of determining when a customer has sent us something that is similar to a known toxin or pathogen. I think some of the current practices might see challenges by some of the AI-based protein design tools. Twist and some other IGSC companies recently published work with Microsoft demonstrating that the current generation of these AI models are sort of unlikely to pose significant risk of being able to engineer proteins in a way such that they reproduce the harms associated with existing pathogens, but don't maintain the sequence homologies, which is what we use to detect them. As those model capabilities improve, though, I think Twist is right now working on developing new defensive models that would help predict that kind of engineered risk in our order streams. Outside of biosecurity, in the security world more broadly, we also really place a lot of importance on the degree to which our customers' intellectual property is encoded in the DNA sequences that they send us. So these represent a lot of intensive work on the part of our customers. And so protecting those resources is incredibly important. And DNA is what we refer to as a self-documenting molecule. So the first thing we do in a biosecurity context, when a sequence comes in the door, is reverse engineer it to understand what it is, where it comes from, what it does, as a means to assure ourselves for biosecurity purposes that there's no misuse intended. But that process also exposes the intellectual property of the customers. And so we act as trusted agents of those customers. But this begins to get complicated when you have deeply international supply chains. And we certainly urge customers to make sure that they have a lot of trust in the partners they're working with when they're sending these materials or with this data for synthesis. So I think I'll leave it there. I look forward to the discussion, but that's kind of the framework in which we think of security around nucleic acids. Thanks.
Great, thank you, James, and thanks to all three of our panelists. I have a few questions that I can kick off with, but I would like to open it up to anyone else to start. I'll give you 3 seconds.
In the room, we have Cameron. So Cameron.
Okay.
Can you all hear me? I don't
Yeah,
think
you sound great.
These cameras do work. I'm Cameron Kim. I'm at Duke University. My question, I think, for James right now, I mean, one of the things that we are tasked to think about are, different types of chemistries or different types of approaches that, individuals might make proteins, right? So looking at non-canonical amino acids and unnatural nucleotides. Now, unnatural nucleotides may not necessarily show up in the Twist synthesis process right now, but when we start seeing, different ribosome coding, different codon, what efforts do you think either you as Twist or as sort of the entire synthesis and sequencing consortium can handle to recognize that these may show up? But then also, what data would you need, or what would you need to know in order to, catch people that might try to circumvent some of the traditional coding rules that we're familiar with?
That's a good question. I think if we find ourselves in a bright, shiny future where there are some limited number of alternative coding strategies, fairly straightforward to add that to the existing screening systems. We already screen in 6 frames with the standard genetic code, so just adding those on is just a bit more compute. So I don't think that would represent a real threat. I think the real challenge is if we get to a place where essentially randomized coding strategies can easily be used by anyone, then we face a challenge in not being able to interpret the protein sequence represented by the nucleotides that we receive. And so there you start kind of going into less reliable methods like frequency analysis and assuming a certain underlying distribution of structural amino acids. And it can give you maybe some insight into what was being used or attempted by the customer, but it will not really give you a clear ability to predict the final protein sequence. So I think that's kind of what we're watching in terms of trajectory.
Thank you. I have one question, and then we can open it back up to the room. All of you kind of mentioned, in a way, innovation and the relationship your individual organizations have with 4S. And 4S for everyone is security, sustainability, social responsibility, and safety, obviously. As we know, there are 2 kinds of people, right? There's always 2 kinds of people. those that would see something like 4S as a hurdle, and those that would see this as a springboard for innovation. And so just I do want to ask all three of you maybe to comment on how we in industry or in industry serving organizations can use 4S to drive innovation and progress, using 4S as guiding principles, not seeing it as a hurdle to commercialization. So maybe we'll pick it up with Lauren. You have innovation in the title, so.
Oh, yes. So thank you. It's a good question. Obviously for BASF, you know, we need to be able to sell our products and work with our customers to be able to have them be approved in whichever regions of the world are going to be, where they're going to be utilized. And so, of course, you know, the societal responsibility comes with that. And so we, you know, there is part of an opportunity for innovation so long as let's say the regulatory framework is clear. Where there's uncertainty, where we could just look at genome editing as one example. In certain parts of the world, this is okay, particularly for plants, and other places it is not. And so this then stifles our innovation because we could bring great products, but oftentimes we will take our innovation forward when there's a global market for it. And sometimes if it's only a regional play, then this becomes more of a difficult decision to put those research dollars behind that initiative. So I think maybe that's where I'll start off, and then I'm sure James and Steve have something else to contribute, and I'll chime in as well.
Yeah, and I think that just tying it back to this, like why we're all here today, maybe something to take note for the committee here is that clarity, because I know Steve also mentioned regulatory, like that clarity around what is the requirement and where. And so thank you for also mentioning it. I'll turn it over to either James or Steve for comment on how 4S can drive innovation, not hinder it.
I'll go ahead and jump in. So I think it is important to think about the difference between innovation and commercial activity. There are, and that's a place that there can be this chasm of relevance. And so what entities need to have are products that are recognized as being safe and effective and not detrimental to the environment and to the communities that surround them. And so there is a commercial reason to be to use the 4S's to drive your commercial development. I think, you know, we can take a simple example. If you really focus on safety in the broadest definitions of safety, and suppose your organization needs to have multiple production facilities, either around the country or around the globe, you can have principles that are used in the design of your facility, the design of your processes, that are easy to replicate and that have been tested and known to be effective in safety. And so then it becomes just a core part of your operating business, and you can really focus on your business practices when you're you know, at multiple facilities, if you're not having to invent it new at every place. And so I think it's an area that can make you competitive because you have a good framework to build on. Over.
And maybe I'll just highlight that a lot of the safety and security work, I think that the nucleic acid industry has been engaged with, has really been about building trust, making sure that these technologies, while they are very powerful, are trusted by everyone involved in that ecosystem, governments and customers and the public alike. And so ensuring that you put enough effort in to understand any security risks and mitigate them, safety risks, mitigate them, I think really is incredibly important in terms of ensuring that innovations can be disseminated and used widely by a public that trusts them.
No, that's a great point, because if no one will buy your product, there is no bioeconomy. I'll
Yeah.
open it back up to Lauren, do you want to?
Yeah, no, I just wanted to circle back to something that Steve said, really around that safety, and having, starting, we like to say, start with the end in mind, right? Of knowing what that product needs to be, what targets it needs to hit. For instance, if we look at a bioproduct that has, let's say, an enzyme, an industrial enzyme, if we know upfront of what the level of let's say, let's just say GM DNA is allowed to be in there, then we can design that process up front. Reshaping that process along the way becomes very expensive. And so again, operating with where there is a clear framework, again, for the safety of that end product, so that we can sell something, is key. That's just one example of, with genetically modified DNA in a certain product. But also, on the agricultural side, safety of those products, is, let's say those crops, is also very, very important. And we do spend a lot of time, effort, and money to ensure that we if we're starting to sell something or even doing field trials, that there's adherence to all of the local and federal procedures and policies. So that's an important piece too. Yeah, thank you. There was a question in the room, I think.
Felicia?
There's several questions here. So, and if you can't see anybody, I'm happy to call them. So, Lauren, it's wonderful because in fact, you answered a question that one of my colleagues and I were just talking about this morning. We would love to hear your general perspectives. You mentioned begin with the end in mind. When you're thinking about developing these new biotechnologies, at what point, whether in research or in thinking about commercialization, do you begin to think about the necessary regulatory hurdles to be overcome, as well as the topic that you brought up? I believe it was James, about public trust and involvement of potential consumers. Thank you.
Yes. So I guess I'll take that one first. So yeah, it is very important that we, if there are regulations, that we, take those into consideration from the beginning. I mean, we won't even start a biotech project or any project without, let's say, a clear techno-economic assessment, of how much will it cost, how much is the feedstock cost, how much is and then of course, then we have a calculation as well of how much the R&D should cost, and the scale up, and what's the market for talking about a chemical, bio-based chemical especially. But understanding also how that product in the end, if it's a bio-based chemical, is it a drop-in, or will it need to have a new regulatory review? This is something that's very important to us, and we have entire departments at the large company like BASF to make sure that we're dotting all the I's and crossing all the T's, and also staying abreast of what those new regulations look like in every region that we want to sell to. If we look on the agriculture side, this is our ability to be commercial, right? So if for instance, Europe decides that they're going to allow or not allow GM crops, this is a big decision for us, or same with Japan or India or China. This is a big decision for us, or if the US changes any view on that. So we are constantly keeping an eye on the regulations and how they're evolving, so that to in some ways preserve our products, to keep them still on the market, but also and understand where they can be sold, but also for new products in the pipeline, and quite frankly for agricultural traits. Yeah, so these would be the GM traits that go into broad acreage crops. This can take 10 years. So we need to have as much clarity as soon in that process as possible. And so it's not unlike a drug, where that time frame to get things on the market. I hope
If
that
I-
addressed your question, but yeah, Steve, please.
Oh, if I could, I was going to jump in and also argue to tie back to the prior question on innovation. You have to understand the framework of regulations in order to actually develop an innovative product and accelerate its development. There's some interesting work that's come out of a group at Arizona State recently that in fact argued that efforts to incorporate intrinsic containment mechanisms actually do not result in products that move into commercialization. And so it's a framing of understanding the nature of what your product might look like and the terms that are important in those regulations, intergeneric and so forth, to then devise the kind of product. And James mentioned something, earlier about the 6-frame translation analysis. Again, if you don't know that your construct to pass a regulatory evaluation in Europe or the US is going to undergo a 6-frame analysis. You get in there and there's this alternate reading frame that you didn't deal with. You now have you've basically shot yourself in the foot for a moment as you move through a regulatory process. So you need to know that early, design that into your process early. And so being very cognizant of those ways in which something's going to be evaluated is important to minimize surprises in your regulatory journey.
And maybe I'll just add to this by saying the nucleic acid industry is only very lightly regulated, primarily for export control. And so it may serve as kind of a counterexample to the more heavily regulated product-focused companies, by which to say a lot of the reason it remains lightly regulated has been a combination of both governments being hesitant to regulate new innovative industries and products, as well as the industry being proactive in trying to carry out, develop practices, and build trust that they're acting in a responsible manner. So that interplay, I think, has been incredibly important for the nucleic acid synthesis industry.
Thank you all so much. By the way, Steve, you were mentioning the ASU study. We happen to have one of our ASU scholars in that area. Also, Dr. Emma Froh is here. So yes, a co-author of that report. You had a question too? Is it right?
Yeah, I think, sorry, this is India Hook Barnard from EDRC. Hi, you guys. I think this is picking up maybe a little bit, especially to Steve's point about the importance of having the shared principles across organizations and maybe manufacturing sites or others. And I'm just wondering, to be able to do that, like right now, you also have maybe an advantage by keeping it close and keeping the innovation, to your own company. But the industry as a whole would do better if we're able to share that and have, shared standards and other things. But that requires the data sharing and basically for somebody to organize so that industry acts as a whole best interest rather than the individuals kind of we kind of have like a prisoner's dilemma or something right now. So I'm just wondering if you could speak to that and about the importance of standards and or data sharing for how to kind of make that happen, or if there's regulation that's needed for any of you, but maybe Steve, if you can.
So I think there are very important areas in which data can be a driver in these, sharing access to data can be a driver in safety and security. I think though that, maybe pre-competitive organizations or organizations of that type can convene and promulgate standards or at least provisional guidelines, right? The idea of this is a good idea to do this, a good idea to do that. And so this is an area that, at BioMade, we try to do through our 4S committee structures, but there are other organizations that I think the challenge is, setting formal standards, especially something that will take you, say, through an ISO framework, is labor intensive and time intensive. And our industry moves more quickly in some ways than those standard setting bodies can operate. I think another challenge in particular for the biotech community is that it really is comprised of a lot of very small companies. And those companies don't have the resources to commit to standard setting bodies. You can't just allocate 2 people, in a 5-person company to do that. And so there are some structural dynamics, I think, that maybe the committee can think of ways to reframe the development of best practices and the promulgation of those. And I guess the last thing I would say is not all of that requires access to underlying data, but figuring out what data may be important, I think, would be a good component of that. I'll stop there.
Well, and Steve, if I can pull on something you just said, it's these small companies. Like Lauren mentioned that BASF is watching globally at all times for any regulatory shifts, and they probably have an entire team or part of the company devoted to do so. And so one thread to pull or one thing to just maybe put in the committee's minds is how are we supporting smaller companies who don't have those types of teams behind them when it is so ever-changing from a regulatory space, and how do we make sure they know the problem they're trying to solve for in the time that it's needed. So just something to it's not really a question, but more of a like putting a pin in that.
Maybe very quickly, though, I think Lauren's company, they would never be surprised to know that they need to file an MCAN. And they would need to know what the components, including the safety components that go into framing an MCAN. But there are many small companies who have a technology idea that they want to try to move forward that don't even know what those 4 letters mean.
Yeah.
And so there's a lack of understanding just because some of it's very deep technically, very with its own jargon. And then when you think about that across multiple regulatory agencies that you may have to go to, it just becomes a challenge for a small company. Over.
And I just wanted to also maybe mention, you mentioned across agencies, this is also where there's possibility for interagency coordination. This would also be wonderful because again, if we talk about say ag tech, biotech products, this could be touching on the EPA, the USDA, the FDA, and maybe some other as well that I'm unaware of. But at least those 3 agencies can be involved in the review and approval of those products.
I just wanted to follow that to James on that question about maybe standards or principles across the industry and data sharing. Especially if you had anything to add on that IP piece as well, because we were also thinking about if and how IP is part of our scope as we're thinking about data sharing and the security frameworks.
Yeah, and I would say we certainly have, the nucleic acid synthesis industry has kind of divided the world up into things we're willing to collaborate on and things we are not willing to collaborate on. So we're all very aggressive competitors, but we still come together in this IGSC forum and talk around how do we reduce the risk of misuse. One of the things we've long worked on is a shared data repository that we call the restricted pathogen database. The governments publish lists of species that are controlled, but we make sequences. And so we have to somehow be able to make that map between sequence and controlled organism. And so the IGSC has created the data resource and shares it across the global membership, 42 members in 10 or 11 different countries. And so that has been incredibly valuable. We've also done recent work with NIST and EBRC in developing ways of measuring how these screening systems that we've all built perform, because it's a really complex kind of human in the loop sort of system where you're producing a bunch of data and then asking a human to make a value judgment. And so how do you measure systems like that so that you can compare across companies? And on the intellectual property side, I think there was a really important addition to the US government guidance in the 2024 OSTP framework, where they clearly called out cybersecurity and data protection as something that industry actors should take seriously. So Twist maintains an ISO 27001 certification, we spend a lot of resources on protecting our systems and data. And that's primarily because we know that we're a trusted steward of a lot of very valuable intellectual property. And that's true across the industry. And so it's really important for companies in the supply chain to realize when they are stewards of high value information and to spend appropriately to protect that.
For your answers. We have another question in the audience from Dave Dunning.
Yes, Dave Dunning, University of Michigan. I just want to follow up a little bit on the question that Felicia asked about regulation. Let's say regulatory unclarity or regulatory clarity. That's come up a lot, and I'm wondering about sources of regulatory unclarity. You've mentioned some, and I wanted to see if I could press for a more comprehensive list. It sounds like there might be unclarity due to different agents, that is, Europe versus the United States, or different agencies of different languages and different rules. There can be unclarity because regulations change over time. And there can be unclarity because you have an innovation or a product that has characteristics that doesn't really fit the framework that's in front of you for regulation. And I was wondering, are there other sources of unclarity, or which sources of unclarity seem to be the most troublesome or the most challenging in terms of bringing something finally to market or to bring a benefit to society? Or what the basic question is, what are all the sources of unclarity that you see when it comes to regulation?
Well, I think you touched on many already. I mean, if we look at Europe as an example, right, where GMO, right, and the definition of GMO versus how genome edited is viewed, right? So there's as that conversation evolves, so GMO has been, a hard go, let's just say, in Europe. But as the techniques with genome editing have come along, this is maybe thawing a bit. And so I think there does need to be, and I think I said this in my introduction as well, there does need to be an evolution of the regulatory framework as there are new technologies that come along. Again, whether it's AI or synbio or non-canonical amino acids, there needs to be a scientifically based approach to evaluating what those regulations look like. Yeah.
I would say, in general, I would say that there is, back to this framework of a disconnect. A lot of technology innovators have come out of a national lab or a university, and they are leading with a technology. And for the moment, let's just say CRISPR. But the US at least doesn't regulate CRISPR. It regulates product. And so when you walk into to a regulatory agent and go, hey, I've got this cool thing called CRISPR, but what are you going to do with it? What is the product that is going to come from that? And so being able to have meaningful conversations when you can encapsulate a technology into a product is very helpful, because the questions are still going to come a little bit about how you made it, but a lot about what is the net safety then of the ultimate product that you have. And so that is it's a different framework of thinking, and it's a different approach to, I think, the question. And so being able to translate what you would do as a technology into the products that could arise from it can help clarify some of the confusion. But there's confusion until you do that.
Is that a conversation then between the scientists, the innovators, and companies, or science innovators and regulators, or all three? Or for those early conversations, who should be involved?
In part, I think it is something that should come into framework even at the university level. And I talk about this in one other framing, which is you can go to major universities and there will be faculty who understand the FDA process. And you can come in and they can walk students through an FDA NDA process. Outside of that, if you go look for other expertise, it's very limited. And so when someone wants to do an environmental application, or when someone wants to do an agrobiotech innovation, they often don't even know who to go talk to on a campus to know what questions they even need to ask. And so I think there is a part of a framing that can come from how we think about innovation in the academies. There is obviously a connection between companies. And so I think, Lauren's company and a lot of other companies have started sponsoring, catalyst organizations, innovation academies. And so I think there are opportunities like that to help bridge that gap. But some of it starts early and just shows a dramatic difference that exists between how biomedical applications have been regulated and moved into the bioeconomy versus much of the rest of industry.
Well, and I think it's important to mention when you talk about going to universities, and there's maybe one professor at some universities that can walk you through it. It starts to bring up, like, where are we training that workforce when any of these companies, including my own, need to hire somebody that's going to do that role within a company? Where are these people coming from, and how are they having to adapt what they learned to apply to this field? And part of it is, should there be a course that is or at least part of a course that can be resourced online, because textbooks are, I'm assuming, gone in universities or on their way out, but having more of an evergreen body of resources so that someone could teach a course to the future employees of these bio economy facing companies. And so just opening it up to maybe James and Lauren for how would you recruit, and how are you seeing that workforce development? And then Steve, as someone maybe to close it out with, how are organizations like BioMade looking to support that workforce development to then be hired into this space? So, James?
Yeah, and maybe I'll just put a quick note in here. I said we're lightly regulated, and that's true, but the main area where we focus is on export control and trade compliance. U.S. regulations there really focus on lists of pathogens and toxins, and then there's language there that says if you have a gene from a pathogen, a bacterial fungal pathogen, and that gene, quote, endows or enhances pathogenicity, then you need an export license. And like from a human perspective, that's exactly where you'd want to draw the threshold. That makes total sense. But from a commercial transaction, if someone buys a sequence from us and we have to make a value judgment about whether it endows or enhances pathogenicity, like that can often be incredibly complicated. There are mechanisms for engaging with the regulator directly, getting a classification. Those tend to take longer than our normal turnaround time commitments to our customers. And so while we do that, it is something that puts certain business transactions in a riskier category in terms of the customer feedback. And on the training side, it's actually, I'm glad you brought that up, because it's incredibly difficult to find people with molecular biology backgrounds who have a background in pathogenicity and are interested in trade compliance and export control and want to evaluate individual orders and the risks they may pose or licenses they may require. So that's certainly not something that we find off the shelf from universities. And I think any kind of educational opportunities that we might think of that would allow folks to develop those skill sets, I think would be valuable broadly in the supply chain for biotech.
Yeah, and I'll double down on what James just said around being those candidates, those people, those folks going into those roles, being able to speak two languages of the technical as well as agencies language. I would say the typical path for folks going into, let's say, that regulatory compliance role, they're coming from the technical side and getting trained up. So it's would it be a benefit for them to have a basis of how to navigate regulations? Yeah, it would be. So, thank you.
Yeah, I would say a thing that is a potentially interesting point in time. If you think about, we just had the anniversary, 50th anniversary of Asilomar, and we have things going on in the federal government, in staff in agencies. There's a lot of talent that is either intentionally retiring or having a job change thrust upon them. And that talent, some of that talent is in the exact areas that we're talking about from these agencies. And so are there ways to harness some of that talent right now in ways that could change how we propagate information? There have been numerous attempts to take the U.S. regulatory agency and put it into an if-then framework. It's challenging. And so having humans in the loop still may be an important component of that. So I think one thing to think about is, are there opportunities with just the state of technology and the workforce to find seasoned veterans from companies and from regulatory agencies that could be tasked with putting together some training. I think, to echo what both of them said, it's hard to find this other than FDA out of the basic university setting. And so getting, a thing that is, I think, important are internships that get people into companies so that they can understand how companies have to move through and act on regulatory frameworks. And just the idea of training broadly at the bachelor's level and even pre-bachelor's level. How do you think about talent and expertise at the technician level, even that could be used in developing expertise base? Over.
Yeah, thanks, Dave. We do have a question online. Felicia, you had your finger up too.
There's two questions.
Okay.
Well, I think Trisha has a question in the room and Chris Cummings. Okay, so Chris.
Thank you. I've been really intrigued by the discussion on regulatory constraints, but also on potential opportunities. And Lauren, you mentioned how there is oftentimes kind of a regulatory gap or kind of a pacing problem between innovation and then regulatory oversight that tends to come later. And a couple of you have also mentioned that your companies are exploring and or using artificial intelligence in ways to potentially maximize new benefits and opportunities for your country or for your companies. And I'd like for you to speak a bit more about what kinds of opportunities you see and what are the challenges in the current lack of regulatory oversight for uses of artificial intelligence in the kinds of work that your companies do. And I'll open it to whoever may like to respond. Thank you.
I can take that quickly because I may have the simpler answer. So we are very interested in using AI in lots of places within Twist, design principally. In terms of the security implications and regulatory implications, we're trying to figure out how we build models to defend against other models. So you have models that are engineering sequences that could make them difficult to detect when someone's trying to create a harmful biological function. How do we build models to defend against that? On the government side, it would be fantastic if we understood kind of what the research agenda was from a federal funding standpoint to support kind of programs to do this kind of biological risk assessment, model driven. And because it's not something, we have necessarily core expertise in. And so right now we're just trying to kind of form a coalition of the willing.
I'll jump in a way that's tangential because I do like insights from other areas. And so I was at a conference, largely staffed by petroleum engineers. And one of the things that they were talking about is deploying AI models inside their firewall. Again, so this is a thing about how companies think, but they have decades of operational data. And then they even have very detailed schematics of their facility designs in multiple locations. So they were using AI to try to correlate facility design features with safety or safety incidents. But because they have data, they could do that. They were also then trying to use the large body of safety incidents and reactions, or plant operations and the reactions that humans then done, to try to correlate and turn that into a body of knowledge that could be transferred to new workers on the one hand, but then also inform possibly real-time operation. So that's an example that, where could data help? Well, they were able to do it, but it's because that individual company has the resources to deploy an AI tool internally, and they had decades of data. That won't fit all of biotech, but by analogy, there may be ways when we think about facility design or such that AI could come into play. Over.
Yeah, and building on what Steve just said, we are one of those companies who has our own, let's say, AI tools using our own data, and there's considerable benefits for that, of course, to accelerate the safety and design of our from thinking about the steel of how a facility is built and operated, but also safety, of course, but also for thinking about biotech. How is it that we can do our systems more efficiently? How is it we can design a protein to be, have the thermostability that it needs to have in a certain process? How is it that we can design a trait? So there's so much potential here, and again, we use this primarily as a tool to develop a product that will ultimately get regulated. So it's so I see right now there's great opportunities, and again, BASF is very cautious on a lot of these things, so we're self-regulating in some ways too. So we are, when we do use any of the AI tools, we have to either get extreme permissions from our IT department, or we have to use the firewall tool, which is of course custom built. So, but yeah, these are very powerful tools that are coming online.
And I guess something to pin for the committee there is the difference between large corporations and small companies, and there could be a gap that starts to grow with the availability of those tools. And so maybe something to just keep in mind. So we have 2 minutes left. I know there's a question online, Trisha. If you could do it in 30 seconds and we could pick one of our folks, or we can close here, because I know the next session starts right at the top of the hour. But I do want to thank. Oh, go ahead.
I was just saying I could make it quick, or I could just follow up online afterwards.
I'll go. Go ahead.
Going back to something that Steve said earlier, so I guess it's kind of a question for Steve, but everyone can weigh in. I was just curious because you said something about regulating the products and versus the processes, and like thinking ahead and anticipating anything that could come down the line in terms of a product versus a process. But just thinking into like one example of the mRNA vaccines, obviously it's a similar process, but it comes up with different products each time. So I'm wondering, like, it seems like there's this tension between like trying to streamline regulatory processes, because it seems like if you were to look at a process and regulate the process, at least somewhat, it could streamline things, maybe reduce uncertainty, but then obviously it seems like there's also pros to thinking about it at the product level. So I'm just wondering if you have any comments on that, if that question made sense.
Definitely, we'll follow up online. My high level point is in the time that it took the FDA to oversee the development of one genetically engineered fish, they approved 100 monoclonal antibody process. And so there's a benefit to the lather, rinse, repeat kind of framework that comes in versus totally novel application. But we can follow up.
Well, one other thing, just to, I know I'm not on the panel, but I'm going to answer it too, is we have to be careful about prescriptive processes, because there's nothing more stifling to innovation than only having one way to do it. And so thinking about an endpoint as well. So there are arguments for both, but if we could get it to a more turn the crank, I think that's valuable, but being careful with, in the biological space, there's so much room for innovation, so many new things that people are discovering every day. And so being careful with that prescriptive process versus solution. So there's a happy medium balancing all of that, I'm sure, but it's just going to take more mental acrobatics on our parts and many of our regulating bodies. Thank you all so much for the active participation and the great insights from the panel. I got a lot out of this. I hope a lot of you did as well. And I will turn it over. So thanks, everybody.
Thank
Thank
you.
you.
Thank you all for that. I guess we're gonna.
So we need to do a little rearrangement for people up front, I guess, so we'll.
Yeah, we'll take another minute to get the next panel set up and ready, so bear with us, and then thank you.
Hey, welcome back, everybody. We're excited for today's session on funders' perspectives of environmental safety and security considerations for engineering biology. My name is Richard Murray. I'm a faculty member at Caltech, and I'm a member of the National Academies Committee on Environmental Safety, Security, Evaluation on Engineering Biology. So thanks to the panelists. We have 2 online and 2 here in the room. So today we'll be hearing from Megan Palmer from ARPA-H, Ernie Glover from the Alfred P. Sloan Foundation, Valerie Martindale from the U.S. Army Research Laboratory, and Andrea Hudson from Schmidt Sciences. So each panelist will make about 5 minutes of remarks, and then we'll have plenty of time to hope for questions at the end. And so with that, we'll just go in that order. So I'll turn it over to Megan.
Thank you very much for the opportunity to be here today and to see many old colleagues. My name is Megan Palmer. I'm the lead for responsible innovation at the U.S. Advanced Research Projects Agency for Health, or ARPA-H. I thought I would share a little bit about ARPA-H, my role and my background, and the how and why of our approach to responsible innovation at the agency. So ARPA-H's mission is to accelerate better health outcomes for everyone. Our agency aims to address health problems that cannot readily be accomplished through traditional research or commercial activity. The agency has a unique structure and position to deliver transformative health solutions. So what does that actually mean? Our investments seek to address seemingly impossible barriers to demonstrating solutions to major challenges, not just incremental advances. The ARPA model also enables visionary program managers, or PMs, to identify and tackle specific problems, working with a range of performers and a variety of approaches to solve those problems. The agency was authorized into law just about 3 years ago on March 15th, 2022. And it has launched many different programs so far, including precision surgical interventions, security for healthcare systems, therapies for rare disease, and much more. It's a really exciting portfolio of activities. So I joined ARPA-H at the end of 2024 as the inaugural lead for responsible innovation. My role is to lead initiatives that enable everyone at the agency, that includes the PMs, but also our mission office directors and our agency leadership as a whole, to proactively attend to societal implications of ARPA-H's investments. This includes how our innovations shape safety, security, and future options for governance of these frontier capabilities. My background, knowing many of you in the room here, is coming from leading and researching similar initiatives to this across the last 15 years across academia and nonprofits and the private sector, largely focused in synthetic biology. Some of these includes a National Science Foundation sponsored engineering biology research center. I know Teresa Good spoke yesterday here, who was involved in that initiative, the International Genetically Engineered Machine Competition, or IGEM, and most recently Ginkgo Bioworks, which is a public company, and I see Christina Agapakis here, who is a former colleague there as well. And I was really excited to take on this new role at ARPA-H to bring the lessons from these types of experiences to drive a compelling and important mission that really can touch the lives of all Americans and far beyond. So I wanted to share a few things that guide our approach to thinking about responsible innovation as the committee considers how these type of approaches can be brought into other settings. These are principles that really guide our overall approach at the agency, including the ways we drive responsible innovation. So the first we've already spoken about here is we're very outcome oriented. So the agency is really focused on identifying the most important problems in terms of lives and livelihoods, but also in terms of maximizing the return on investment of our precious public dollars and on identifying problems that others can't or simply won't solve. So this is the most important element. It is the central challenge of how we choose those problems that we want to work on. Secondly, we really strive to proactively anticipate what else is needed for our investments to deliver the types of solutions that we need, again, for all Americans and beyond. ARPA-H has an entire mission office that's focused on transition needs. And we seek to engage really across a very complex ecosystem. So this includes work to anticipate where our innovations may be in need of changes and advances in regulation and guidance around it, and overall governance approaches. ARPA-H can create unique incentives for performers to participate in public-private partnerships. And we also have a unique authority to reimburse the FDA for pre-competitive activities. This speaks to some of the other types of questions that we were addressing in the last panel. Third, we strive to be both future-focused and people-centered. So we seek to actively engage stakeholders in shaping our programs. We have a network called ARPANET-H, which is a growing nationwide health innovation network that are anchored by three regional hubs, including one focused on customer experience, and really understanding some of these present day needs and challenges. But we're also, again, relying on these visionary program managers that can see the future, can anticipate a better way of operating that doesn't protect or marginally ameliorate incumbent approaches that aren't necessarily meeting, again, both the challenges of today and those we anticipate to be growing in the future. Finally, we are structured to enable active management and accountability. This is really important. So as opposed to other agencies that might be awarding grants, we use our statutory authority to enter into what we call other transactions, or OTs. We also use traditional types of financial assistance tools like cooperative agreements. Our programs as a whole are also coupled to this evaluation and measurement, and the expectation that funding will not continue if performers do not perform. So that creates the types of mechanisms that we need for an enhanced oversight, not to slow down, but overall to stop activities that aren't working, to ask the question of what working means and to speed up and foster what is. So I've got a little long here, but I just want to give one additional tactic that helps to create some detail for how we operate in practice. A tool that we use is called the Heilmeier Catechism, which is a series of questions that every program manager needs to be answering to justify their programs at multiple levels. This list is adapted to go not just asking what challenge are you going to solve, but asking like how your programs in practice will maximize potential impacts, how you will engage stakeholders, and how they will mitigate potential misuses and harms and misperceptions. Now, we take these challenges really seriously, and we expect that the program managers will have strategies reflected in their program designs to address them. And as we've reflected in other panels, this is not an add-on, but it's embedded in the central goals of the activity and what is resourced in the types of budgets, the teaming, and partnerships. This means that we have metrics that we hold performers and program managers across the agency to demonstrate the need to, for example, show the reliability of a fail-safe design in different settings and in conjunction with the types of regulatory bodies and standards. So I'm just going to end by saying, as somebody who's studied these issues across many different places over time, and the ways that policies and practices manifest, I am deeply aware of the central challenge of figuring out what it means to succeed or fail in these types of challenges. And so what I'm excited about at the agency is we're embedding this central challenge of defining success and failure at every level in the agency through our responsibilities shared across different levels, and also having our programs that take up as part of the central problem, the challenge of defining safety standards, future governance approaches, and more. I'm happy to share more about that, but I just hope that many in this room and those listening will join us in that challenge of defining those standards and putting them into practice. Thank you very much.
Great, thank you, Megan. Next up is Bernie.
Hey, can you hear me?
Yep.
Is that a yes or no?
That was a yes.
No? Can you hear me? Okay,
We
great.
can.
Good morning. I'm Ernie Glover. I'm up. Okay, that was good. Thank you. Okay. I'm a program director at the Alfred P. Sloan Foundation in New York. So I'll start by saying that the Sloan Foundation makes grants across a wide range of areas that include science, higher education, economics, and technology. You know, we do support synthetic cell research, and we do that through our Matter to Life program, which is a broadly scoped program that aims to advance understanding of simple living systems. So start with a few words about funding priorities. I'm going to start with the obvious. Any single private foundation operates with a relatively small budget and therefore supports a narrow range of science compared to what's supported by the agencies. So all this to say that unless biosafety and biosecurity is specifically chosen as a funding priority, then a program director like myself will be under some pressure to constrain biosafety grant making so that it's a modest component of my overall grant making portfolio. So one reason I raised this point is that in the past, before I arrived at Sloan, Sloan had a program aimed at biosecurity that made grants to help prepare the public for bioterrorism and related threats. And that program ran for about a decade. It ended in 2010. And our current Matter to Life program mainly supports basic science research, so it's very different in character from a program that's dedicated to biosafety. So that said, the Sloan Foundation does indeed want to be a responsible science funder. And in particular, we want to do what we can to support socially responsible development of synthetic cells. So I'll share a few thoughts I have on the challenges and opportunities I see associated with that goal. First is a challenge. There is a need for research to better understand today's biosafety risks and how to mitigate them. But as I've mentioned, I'm constrained by the number and the size of biosafety grants I can make. And supporting biosafety research would be much easier for me if the research tackled, fundamentally interesting basic science questions, since that's what my program is really aimed at. So to the extent that proposers can frame their biosafety research as also exploring interesting basic science, it'll be easier to fund, at least from my perspective, it will be. As a related point, the LC Review Committee is charged with making recommendations about the biosafety research agenda. And it's important that NSF is sponsoring this study because I imagine that's going to be a pretty ambitious agenda. So NSF and NIH will have to play significant roles really driving that agenda forward. Also, it's important to understand, what's going on globally with regard to defining a biosafety research agenda, since the risks are global ones and some coordination will be necessary. In another direction, one of the opportunities I see for Sloan
Hope he comes back.
Yes.
We're sorry, you're cutting off a bit.
You're good, just a little bit. When he comes back, we'll let him know. Is it?
Yeah.
Other gathering different.
All right, we'll give him another minute just in case he comes back so we don't lose the thread. And if not, then we'll come back to it. Yeah, I imagine he's going to relog back in with a little bit like he was getting whatever Wi-Fi he gets.
Video.
Okay. Right, so Ernie's they're gonna do a little bit to see whether we can't get a better audio for that. So maybe what we can do is we'll pick him back up from where he left off, or try to. And so maybe we'll move over to Valerie Martindale.
Good morning. Can you see and hear me?
We can.
Yes.
All right. And if I need to turn off my video, let me know. So I am the extramural competency co-lead for biological and biotechnology sciences for the Army Research Laboratory. And what this means is that I manage a group of people who fund extramural research in this area. We cover over 100 different universities with active research agreements right now, including some in a number of foreign countries. We have a relatively small budget compared to the big players like NIH and NSF, and we do need to focus on Army priorities. However, what we do is fund basic fundamental research. So while we need to be able to imagine an Army application for it, we do not fund it for application's sake. We fund it for the basic research sake. Now, I want to put some context on this, particularly for the questions that you're asking about safety, security, and environmental consequences. How I like to frame this is we want to be able to enable engineering for fielding. So I'm going to go through those in reverse order to explain the context. Fielding is the goal. Ultimately, we need to be able to use the bioconstructs that we will produce as a country, as an industrial base. We need to be able to use those in the real world. In order to get to that stage, we need to be able to engineer for fielding. So an engineer should be able to look at a bioconstruct on the shelf, so to speak, and pick it up with as much confidence as they might pick up a transistor or a diode, that it would be that well characterized, that the failure modes would be known. Obviously, safety, security, environmental concerns are a very big piece of this, and we're not ready for that yet. We need to look at more fundamental research, go back one step prior to that to enable engineering for fielding. One example that I'll bring up is what I call predictive immunology. The FDA uses trial and error to determine whether a medication is safe. It's the gold standard, and I very much appreciate that they do that. But we should be able to improve our chances of getting through the trial and error process by having better predictive methods up front. And the kinds of things that we're envisioning are going to come in contact with the soldier. If we're talking about uniforms that can sense or respond to something in the environment, if we're talking about water purification systems, there are many, obviously, beneficial uses that we imagine for bioconstructs. And in order to make them reliable and predictable and robust, we need to solve some basic fundamental problems on predicting how they will react with the environment. And so we do actively fund in this area because it is still basic research, but with very clear application, and not just for Department of Defense, but across the board. We're going to have to be able to solve these problems in order to achieve the benefits that we imagine from this system. One other thing that I'll mention is some of the tools that we have available. Our primary one is what's called a single investigator grant. It does not mean that there's only one investigator, but all the money goes through one investigator. We also have the MURI, the Multidisciplinary University Research Initiative. This is 5 years at $1.25 million per year. It must be multidisciplinary. Almost all of them involve a 2 or more universities to collaborate on the problem. And so it's a way to bring a lot of brain power from multiple disciplines to bear on a problem. We also have the ability to do small business initiatives, and this is a way to explore commercialization or proof of concept early in the process while it's still a basic research problem. And I think that is all I had planned to say. So thank you.
Great. Thank you, Valerie. Okay, Ernie, we see that you're back. And so we locked you around, the approach we take at Sloan is something around there.
Okay, sorry. Hopefully this will work. I switched to my phone instead of the hotel Wi-Fi, so hopefully that'll work better. So at Sloan, we do support synthetic cell research at Sloan, and we do that through our Matter to Life program. That's a broadly scoped science program, really aimed at understanding simple living systems. So, a few words about funding priorities. So any single private foundation like Sloan really operates with a relatively small budget. And so we support a relatively narrow range of science compared to what's supported at the agencies. So all this to say that unless, biosafety or biosecurity is specifically chosen as a funding priority, then someone like myself is going to be under some pressure to constrain biosafety grant making. So that's a relatively modest component of the overall grant making portfolio. I will mention that before I arrived at Sloan, the Sloan Foundation had a program that was aimed at biosecurity. That made grants to help prepare the public for bioterrorism and related threats. That program ran for about a decade, and then it ended in 2010. Our current Matter to Life program mainly supports basic science research. So in that sense, it's very different in character from a program that's dedicated to biosafety. That said, we at Sloan do want to be a responsible science funder. We want to do what we can to ensure socially responsible development of synthetic cells. So I'm going to share a few thoughts I have about challenges and opportunities with meeting that goal. First is a challenge. There is a need for research to better understand biosafety risks and how to mitigate them. But as I've mentioned, I'm constrained as to the number and size of these kinds of grants that I can make. And supporting biosafety research would be much easier for me if the research tackled fundamentally interesting basic science questions, since that's really what my program's aimed at. So to the extent that proposers can frame their biosafety research, as also exploring interesting basic science questions, it'll be easier to fund, at least from my perspective. Is a related point, the LC Review Committee is charged with making recommendations about the biosafety research agenda. And it's going to be important. I see that NSF is supporting this study. That's important because I imagine that's going to be a rather ambitious research agenda. So NSF and NIH will have to play a significant role really driving that forward. Also, it's important to understand what's going on globally with regard to defining a biosafety research agenda, since the risks are global ones, and so some coordination will be necessary. In a different direction, one of the opportunities that I see for Sloan and other private foundations is the opportunity to support workshops and other gatherings that bring together different stakeholders to discuss biosafety. I know that the LC Review Committee is familiar with the Spirit of Silmar meeting that was held earlier this year, and I'm optimistic that the reports from that meeting will be useful resources for the committee. More generally, biosafety landscape will evolve as new research creates new threats. So there's going to be an ongoing need for meetings to evaluate the safety threats and to update the research agenda. And support for those meetings is one way that Sloan and other private foundations can contribute. Another challenge is that funders can't realistically take on the role of determining which specific guidelines that should be used to ensure biosafety. At Sloan, we naturally rely on the safety infrastructure at the places where the research is being conducted, so at the universities. That said, we are thinking about what else we might do to promote safe synthetic cell research. So as one example, we recently made a grant supporting research that works towards engineering a translation system that makes use of recoded codons. So in evaluating that proposal and talking to the PI, it became clear that as that type of research progresses, it could evade the existing safety infrastructure at universities. And what we settled on was that the PI would perform an analysis that considered the biosafety implications of the research, specifically focusing on whether and how the research might evade the existing safety infrastructure. So the PI in that case will make recommendations for how existing safety regulations or policies could be extended to cover new threats that are associated with the proposed research. So at Sloan, we're thinking about generalizing this safety self-assessment process by asking grant applicants to include a biosafety plan as part of the proposal submission process.
So the overarching goal there is really to have the PIs give some thought as to how their research intersects with biosafety concerns, and to then discuss a plan to address those concerns when they arise. So this plan would speak, both to risks that are familiar to a university safety committee and to new ones that are raised by the emerging research. So we'd ask proposal reviewers to evaluate the safety plan, and it would make sense to have a safety expert as one of the reviewers. So I'll stop there and happy to answer any questions.
Thank you, Ernie. Okay, and last up is Andrea Hudson.
Hi, everyone. Good morning. Thank you all for the invitation to join you all today. So I am Andrea Hudson. I'm the Director of Biosciences at Schmidt Sciences. We are a philanthropy that's focused, founded by Eric and Matthew Schmidt. We have 5 major focus areas. We fund a range of basic research to use-inspired research across AI and advanced computing, climate, biosciences, astrophysics, and a group called Science Systems that thinks about how science is done and the mechanisms around the sort of ecosystem of science. When we meet within biosciences group, our particular focus is interested in exploring how we can harness the power of biology in an interdisciplinary way, so convergence with other disciplines, to provide public benefit. The majority of our programs so far have been really focused on enabling a resilient circular bioeconomy, with a focus on future feedstocks, sensors, biological sensors for monitoring fermentation reactions, essentially. And innovation in bioreactor design. So really a strict focus so far on biomanufacturing. So thinking about sort of contained use products of biotechnology. A little bit further away, I think, from the major considerations of this group so far. We are actively scoping and landscaping new areas of interest for new programmatic areas for our focus. One of them, I think, will be a particular interest in this group. We hope to get off the ground is that we are currently sort of sketching out the parameters of that. We'll be thinking about the environmental, sort of addressing some of the foundational knowledge gaps around the release of engineered organisms into the environment. And so there we would really like to take into account in a more formal way some of these considerations around the safety, biosecurity, and environmental implications of release, but trying really not to address a particular technology or application area, but probably mostly focused on addressing basic knowledge questions or foundational questions around persistence, dispersal, environmental interactions. What does it mean when we see changes to the microbiome in the local area if we're talking about microbe that would be released? So while we don't have formal mechanisms within sciences to address these questions, it is something that our team takes into account when we review proposals. We think about everything from the reputation of the PI, their experience, and what they have done. Do they think about these questions? If a proposal were to be in front of us that we think could raise particular questions related to safety or security, we would ask those questions and have that conversation with the PI. We'd expect our reviewers to comment it, but it's not necessarily a formal part of our overall process. And I think the last thing I'll lead with is I think we would be interested in learning more about what are the right types of questions and formal processes and leading on us in the space as well to learn more from there. Where they have institutionalized sort of a formal context so that we could be in good partnership, because we do take our responsibility as a funder really seriously and understand implications, even though philanthropy, as my other colleagues have said, is a small piece of the funding puzzle. It is still an important place for us to be active. So with that.
Great, thank you. Okay, fantastic. So thank you all for the comments. And with that, we will open up to the committee for any questions. Go ahead, Cameron.
Hi, everyone. Cameron Kim from Duke University. My question goes to Ernie, actually. You discussed the anecdote around one of the grants that you provided for sort of recoding and sort of the conversations back and forward that you had as a program manager. One of the things that I think we are tasked with this on this committee is how to have scientists and engineers think about this before it gets to your desk and to everyone's desk here in terms of funding agencies. So I think one of the, maybe 2 questions to this for everybody is, what would you like for investigators to have prior to even providing this grant to you so that we know that they are thinking about responsible innovation frameworks in the process of development and design? But also, are there particular responsible innovation frameworks that you all have leveraged in reaching out to people so that it is sort of a known standard in granting processes, something like what OECD may produce or other organizations related to innovation in the space.
Yeah, so it sounds like everybody.
Yeah, I'd say I'm not well aware of what the other frameworks are for ensuring safe research. As I've mentioned, we're not really in a position to sort of think about the specific guidelines that really should ensure safety for synthetic cells or other areas of research as well. So we really do rely on the safety infrastructure at universities. But in terms of what I would like brought to me, really before people start their research, it is really this thing that I've mentioned, this idea that we're thinking about asking for a biosafety plan, which is this exercise of being thoughtful about what the research, what threats the research might raise, and then writing a description of what those concerns might be and how they would be addressed. So this is something that we're really newly engaged in. The grant that I talked about was made, half a year ago, so it's still relatively new. So we're in the process of trying to develop this requirement and think about what, exactly how far spread it should be in our grant making. But I do think that, because we lack the expertise at the Sloan Foundation to really impose specific guidelines, really the most important thing we could do is ask proposers to really give thought to the biosafety implications of their research, put that in writing so that other people, reviewers and biosafety experts can read what those thoughts are and can give us feedback about whether they're taking appropriate measures. So I do think this idea of having a biosafety plan that encourages researchers to give thought to that and to put something on paper and about how they would deal with that, I think that's really where we are right now, and I think that would be quite effective.
Great, maybe we can do in the room and out of the room and go back and forth. So maybe Megan, then Valerie, then Andrea.
It's a good question. So referring back to some of my comments before, our primary power and position as a funder is to be able to provide the types of incentives to pay attention to these issues, right? They're embedded in the challenges we put forward centrally in our programs and the specific metrics that drive our work and by which performers are evaluated. So those types of incentives and the resources that come behind them are one of the key enablers. We also have to ask through our programs in designing them, are there standards, best practices that we can apply? Or is there an area where standards development is needed? As a funder of frontier capabilities, we can reasonably anticipate that we will have capabilities where that standards development is needed. And that is why we have these unique authorities and positions around developing partnerships and being able to fund pre-competitive activities with relevant regulatory agencies, the FDA in particular in this case. And then overall, health challenges means safety is foundational. And we're dealing with challenges where individuals' safety is already at risk through the diseases that they face. And so that is a central challenge. And I'm excited that the agency is really able to not just take this compliance posture, but really help to drive innovation around issues around safety and governance and beyond.
Great, thank you. Valerie?
Yeah, so tying in with things others have said, as Ernie said, we do rely heavily on the safety programs in the universities or small businesses that we deal with to lay the baseline for that. And interestingly, listening to Andrea, there's a difference when you are dealing with an essentially healthy population. We are very conservative in how we think about safety and security because we are not dealing with treating disease where one might be willing to take greater risks, but we are dealing with things that will be in contact with a healthy population. And so the initial standard is very high. for anything that we would even think about. The Army Center for Synthetic Biology, I think, has some framework in place for thinking about how they would get to a test step that is outside of the laboratory. But in general, we have not done that. And again, we have something of a luxury in that we can be what we call fast follower. We don't have to be the first to do this. We can look at other organizations that have a different risk acceptance profile and say, this form of encapsulation or this form of oxytrophy or kill switch or the various mechanisms for preventing spread or release. A personal topic of mine is how to prevent genetic drift in a population. These are things that we want to have clearly stated before we would consider a testing that gets to the borderline of the laboratory environment, let alone outside the laboratory environment. I think that covers the Department of Defense attitude on that.
Great. Thanks, Valerie. Andrea?
Yeah, to address the first part of your question on how do we get scientists to think about this before proposal comes to any of us. I really like the discussion in the prior panel that was getting towards training and raising these issues early on in graduate school training. I think that there's a really good opportunity there. We do this a lot, thinking those that are oriented toward biomedical degrees. I know for myself in school public health, it's surrounded by considerations of safety and security. But I don't get the sense from colleagues who went to school that were focused on generalized biology programs or plant science or other areas who had access to the same molecular biology tools and training, not necessarily getting it. I think there is an opportunity there when we think about our responsibility conduct, research integrity classes that tend to focus on plagiarism and other things that you could expand that focus and bring it, I think, into these other areas so that you can start to embed that within the training, that it is part of what everybody approach to development for their research proposals. So I feel like that there's a unique opportunity there, and I would love to see that consideration. And I think we would, if we were to see a proposal that already had sort of that forward thinking about safety by design, security by design, sort of understand that this is an individual thinking holistically about the implications of their work, and that would make us more excited and more confident about the.
Jason, also the question. Then would you, Jason, then Felicia?
Thanks. A lot of your comments are making me think about the difference between being confident or relying on established processes for sort of detecting safety and security issues as they come up versus adapting those processes or having novel procedures for emerging technologies that disrupt the patterns that we've seen before. And so maybe I'll ask Megan first, but other people may want to comment as well. You were talking about the series of questions that go to PMs and how well do they adapt to new technologies that might raise other kinds of issues versus, thinking about for something like synthetic cells, which might present a different set of issues than what we've seen before. Do we need a new set of guidelines that have new technical components that would help funding agencies think about novel risks in different ways?
The Hammer questions, which were developed and used within the context of DARPA, the Defense Advanced Research Projects Agency, also used across other settings and modified at ARPA-H to include 2 additional questions. They're very general questions, but they allow a lot of specificity underneath of them that allow for the articulation of this types of goals that we've articulated here. Of course, they're not the only tool that we use, and agencies also, of course, have to comply with many other policies that attend to particular issues, especially where there, again, are known standards and policies for which there is always a degree of question around how to apply them. But I think it allows us complementarity of giving the ability to ask a number of questions and give program managers and performers and others the ability to be innovative around their approaches, while also ensuring that we're attending to a wide range of existing policy directives and requirements.
Can I just ask a follow-up on that? Because you mentioned that you've added 2 questions. What are the 2 additional questions?
So there are questions that specifically ask how you will reach everyone, central to our mission. So those types of answers are already present in what challenge are you trying to solve, your earlier questions, and how does it relate to incumbent approaches, but it allows us to be very specific in addressing that central mission. We also ask specifically around potential for misuse and misperceptions. Again, this is reflected in earlier types of questions around the risks that a particular capability will raise, but it allows us to be, again, even more specific about the how and the why. And so those questions, they're online. A program manager, a potential program manager needs to answer those questions as part of even their application to the agency. They come up in many places, but you can see those.
Great. Thanks. Other thoughts on Jason's questions from anybody?
Yeah, I'll just comment that, I do think, particularly with respect to emerging research, emerging threats, really the researchers are the ones in the best position to sort of identify what the threats are, because they have really the technical expertise. We don't have that at the Sloan Foundation. It's, maybe larger agencies can bring in some of that technical expertise in-house, but even so, there's always going to be emerging frontiers where you're not going to know really the right questions to ask. So, it really there has to be a significant input from the research community to identify what those threats are. As I've said, a program like mine, which is really focused on basic science, can help by doing things like supporting meetings and gatherings to bring that community together to surface those questions. But in terms of playing a more direct role, the example I gave before, where Sloan had a biosecurity program, that's a different kind of approach, but that's not the approach I have for my particular program. So it kind of depends a little bit on the nature of the program that you have. But either way, I do think it's critical to really engage the research community in identifying what those threats are going to be.
And if I can speak for Department of Defense, the Tri-Service Microbiome Consortium and the Synthetic Biology Community of Interest both have ELSI panels or functions which discuss these at least annually, and each service has representatives who are active in that, and that also includes the legal and contracting function, which we rely on for some of our safety and biosecurity also. And this grew actually out of genetic privacy concerns early on in human subject use, but it rapidly became clear that we needed much more dedicated and targeted ethical, legal, social, environmental issue responsibility with respect to synthetic organisms and bioconstructs. And so it is an ongoing, continuous thread, I would say, in how we approach the field.
Andre, anything you want to add to this particular question?
I think my colleagues on the panel have addressed the various parts of it. I think the only sort of non-redundant thing I would add is I do think it is the responsibility of those who are sort of funders outside of government to be monitoring and looking for these types of considerations or implications to our programs. I don't know that we need to be, as Ernie was saying, it's not at the forefront of the reason for the program existing. Then I think it's sort of ancillary for us to be aware where there are potential touch points, where there are those considerations that come into effect, and where to seek that type of independent guidance, but not necessarily to be shaping it if that's not our mission.
Okay, I had Felicia, then India, then Emma.
Thank you all for your very helpful presentations. I'm Felicia Rowe. I'm from Michigan State University and a co-chair of this National Academies Committee. I'm very curious. The question was originally for you, Megan, and I realize that there might be answers that everybody here could provide, and that is specifically, what foci of help do you have within ARPA-H and within each of your areas? Currently, I have NIH funding, and one of the first principles we learned was, oh, it's very much focused on the disease, the cardiovascular disease, cancer, lung disease, etc. For your various different programs, are you focused on health effects that might emerge from a technology? Or is it that the health endpoint is an outcome itself? So I'd love to hear your thoughts.
The agency mission is quite broad to, again, address the most challenging health solutions for all Americans. And we have 4 different mission offices under which our programs are organized. Health Science Futures is looking into those frontier capabilities. We have our Resilient Systems Office that's looking at how we, for instance, deal with issues around the safety and security of our healthcare systems and how to make sure that they are resilient. Our Scalable Solutions Office, which is looking at how we actually develop the capability for these solutions to reach all Americans, that scaling challenges. And then the Proactive Health Office, which is around how do we ensure that we'll never become patients. And in terms of this anticipatory posture, how do we make sure that we're solving the types of problems upstream? And then I mentioned the last office is the Patio Office, which is our transition office, and that's really focused on those transition capabilities across our entire portfolio. So we actually really look beyond only one disease. We're certainly tackling many diseases, but asking in terms of that better way, is there a type of platform capability that can address across many different types of disease states? Or is there a infrastructure issue across, again, say the delivery of multiple different healthcare solutions? So we, of course, have to justify that our investments are complementary and doing something different than the other funding agencies.
So others want to comment on, sort of a technology focus versus an endpoint focus. I think that would be the generalization of the question.
Jeffrey? So our current program is not at all focused on human health. So we are interested in sort of using biology as a technology and technology to create products and really focusing on both type of research developments, technologies, tools that would then help the biomanufacturing, community. But when we think about, or as I'm thinking about the landscaping our potential new funding programs, I think there we would probably think about the health of the ecosystems, environmental health, and I'd be interested in questions that would help us characterize what does that mean. What does it mean to see a fluctuation in when there is a release of, for example, a generic organism into soil, right? And how do we understand that dynamic change? And what are those outcomes that we should be measuring for? And so those are the types of questions that we can create within future programs.
Speaking for the Department of Defense, we very much separate medical and non-medical. It is in law that no money that Congress sets aside for medical purposes may be used by operators for potentially offensive purposes. So these two things are kept very separate. If you're a biologist, you realize this is a rather artificial distinction, and I've spent my entire career with one foot on either side of that divide. One way that we deal with that is if we get proposals that are appropriate to a health outcome, we pass them to the Department of the Defense Health Agency because they also are research funders. We do fund research in the area of resilience, of stress response, of rapid medical response in the field. That is the technologies that enable that rather than the health outcome. So if your interest is a health outcome, DoD services are not the funders. It would be Defense Health Agency that would be the funder, if that answers your question.
Yeah, no, I'll just sort of say the obvious, which is, the Sloan Foundation doesn't fund biomedical research. So in that sense, we don't have a health focus, and we're not, our particular program right now, A Matter to Life, that's the one that supports synthetic cell research. And that's not, necessarily, focused on technology. It's really, trying to understand basic living systems in that, sense by trying to build one, a simple living system. So in that way, we're not directly focused on health. It's mainly that we're trying to do what we can to make sure that our funding of basic science doesn't create new health risks. And so as I've described, that's really what we're trying to do.
Thank you all. This is very helpful.
India was next, I think.
I have what I think is specific for Megan, and then if we have time, I have a general one. So when you mentioned you have kind of a unique or specific authority in pre-competitive space, and particularly with FDA to work on those things, are you, I mean, I know in terms of priorities and limited funds that you might be focused in FDA, but are you allowed to work on things that would be would include like EPA or USDA, like if it was the genetic drift questions, antibiotic resistance, and it had to do with like more food and nutrition related to health? Are those things where you would be able to also work with like maybe regulatory agencies more broadly, or are you by statute limited?
So we are statutorily enabled by that unique authority with FDA, but of course are working on many multifaceted challenges that we anticipate will engage with other agencies and groups. I'm also working with our Division of International Affairs that looks at the way that these might touch global communities and markets. And so part of our capacity is to help program managers, again, the entire agency, proactively look at what those relationships and needs might be, and ask, will those types of governance needs become rate limiting in development of solutions? And are we in a unique position to help advance relationships or innovations that are needed to eventually deliver solutions? So that is sort of built into our overall posture, but that unique authority is recognized because FDA is engaged in many of the things that we work on.
Maybe I'll reframe it a little bit to ask Valerie, just in terms of interaction with regulatory agencies, is there any interaction on the DoD side specifically with regulatory agencies, or the types of special authorities that would allow you to work, kind of a special way with a regulatory agency?
Well, this is another area where it's important to say that rather than being at the forefront of fielding something, we are fast followers. The research that we do, we hope, will inform regulatory agency decisions and even the formulation of regulations, that is, how to frame the problem. But it is not typically a direct conversation, and we always, of course, accept the public policy as governing our research and our outcomes. Does that answer your question?
Now, I don't know, Andrea or Ernie, whether you want to say anything, again, just in the context of regulatory agencies and stuff you do with them. I'm guessing not, but I'll let you answer.
I would say not formally, but we do talk to a lot of folks when we do our landscaping, and when we think about who are the right reviewers. I think we have some challenges with getting, direct regulators or regulators to be service reviewers and work with us in that sort of direct way. But we certainly rely on the expertise of folks to inform their do. And I hope that sort of filling in knowledge gaps is the type of enabling information that would be useful to them. I think our ultimate goal with all of our funding programs is to provide sort of service of public benefit. When thinking about environments in place of trying to pay sort of engineered organisms, I do think that is one of my goals would be that it is sort of helping to address some of those foundational questions in a way that is sort of application agnostic. So it's not the role of companies maybe to fill in those foundational assistance in our and so I think we would want to engage with them and hear from them, like what would be most useful and how could we start to help to sort of advance their needs in a sort of indirect.
And Ernie, you may be the furthest away on this particular one.
Yeah, I'll just say, to kind of repeat what I've said a little bit. Our interface with regulations is really to ask the basic science researchers that we support to think about how their research, their emerging research, could evade existing regulations, and to, write a thoughtful, summary of, at both at the beginning of the project, what they think the problems might be, and at the end of the project to write a summary of if they have ideas about how regulation should be expanded to encapsulate the new threats that are posed by their research, to write about that in their report. And then, we'll have to think about how to collect those and make those usefully available to some regulatory agency. We're so early in our thinking about doing this, but that's the role that I see for us as supporting basic science.
Okay, Emma.
Yeah, thank you. So I think this is probably initially at least a question for Megan, because and it's picking up on a couple of things. So I think you have this interesting vantage point of having worked and studied responsible innovation in both kind of academic settings, but also then worked in sort of nonprofit and more applied settings of how do you actually do this work in practice. And I was just struck by the conversation we had in the last session about the disconnects between some academic research and then kind of the world of industry and practice. So just very curious for your take as we're trying to scope out possible research agendas, for example, in biosafety, biosecurity, environmental, do you have guidance for us about, through your experiences, what you've seen and gaps that you see, or, kind of models that we could work productively with, to sort of really try and advance an agenda that helps to bridge some of these gaps?
Thanks for the question. I'll speak personally first on, again, I alluded to why I chose to come to the H from these other types of roles. And it was many of the challenges that our other panelists yesterday and today spoke about are the challenges that we face primarily in terms of the incentive structure and the positionality of different actors working in this ecosystem. Right? Small companies trying to figure out how to use their resources well, these challenges, folks who may have less experience in industry, or perhaps they don't, they've never met a regulator before. And so what I appreciate about working in a agency, especially a new agency, is we're uniquely positioned to be putting these public dollars at work to solve what are often market failures, and in being able to, again, create those incentives to bring together partners that wouldn't naturally come together, perhaps, or to specifically identify the types of pre-competitive needs and activities. And because we are very outcome oriented, again, we look to that future to say what is going to be needed to deliver on our mission. And when we work back from what is needed to deliver on that mission, we see that these issues around the options for governance that can facilitate innovation and the meeting of outcomes, while also, again, we're dealing with people whose safety and trust is also in the hands of these innovations and our potential partners. And our transition office, again, is looking at, we only are part of the puzzle. So we're looking at right from the start at what are the companies, the agencies, the other nonprofit groups, what are their capacities, and how do we set them up for success?
Great. Well, I think with that, we're pretty much at time. And so, in fact, almost exactly at time. So I'd like to thank the panelists very much for your helpful thoughts and insights.
Thank you.
Bye-bye.
Okay, and I think we're going to take a 15-minute break in the room. So we'll pause the webinar and we will be back at 11:15.
Yeah. Hello again, everyone. Welcome to our next open session. And for this, our open session will be moderated by our committee member, Chris Cummings. And so I will hand it over to you, Chris.
Great, thank you. Welcome, everyone. We're excited for today's session on public trust and perception, informing safety, security, and environmental risks. My name is Dr. Christopher Cummings, and I am a research social scientist with the United States Army Corps of Engineers, and I also hold an appointment at North Carolina State University's Genetic Engineering and Society Center, and I'm a member of the National Academy's Committee on Environmental Safety, Security, Evaluation on Engineered Biology. Thank you, first of all, to our wonderful collection of panelists who have volunteered to present, and also thank you to all of those who may be tuning in online to watch today's session. We have three tremendous speakers to hear from today. Michael Zrukian, Professor Emeritus of Medicine at Michigan State University, Dietram Scheufele, University of Wisconsin-Madison, and Christina Agapakis, formerly of Ginkgo Bioworks. Each panelist will have around 15 minutes of remarks, and then we will have time for Q&A at the end. So with that, Mike, I'll turn it over to you, and thank you for volunteering to speak with us today.
Thank you very much. It's my honor. Let me go ahead and share my screen. It says sharing is not turned on, so I think I'll send a request. So if you can let me know when you can see my screen.
I think our team's working on it.
How are we doing? Looks like
I think we're there. Now we're good.
Okay.
Thank you, Mike.
Perfect. Okay. So good morning, and thanks for the opportunity to speak with you today. I'm going to go ahead and share some perspectives from my vantage point as a primary care physician, internal medicine specialist, clinical informatics specialist. I work on building clinical best practices into electronic medical record systems, but I also have a background as an immunologist, a health executive who helped with the COVID-19 response and vaccination effort in Michigan and all the issues related to the synthetic biology aspects of that. Also have served as a professional society leader across health information technology and internal medicine. And I also work as somebody who is part of the public policy conversation across multiple organizations. And my hobby in my semi-retirement is climate and health. So this is a particularly interesting group to talk to from that perspective as well. My focus today for these 15 minutes is going to be to try to summarize the survey data, hopefully to help set up the other speakers as well, regarding what we know in the US and outside the US about public awareness, perceptions, and the perspectives the public has regarding engineering biology as I've been able to identify it, including synthetic cells, and hopefully that'll help inform the research, development, education, policymaking, et cetera, that we're all, I think, interested in. So as a physician, the first questions that patients ask me when their health is at stake, I think matches what we're talking about here very well. First and foremost, is it safe or harmful? What could I expect in the way of benefits, risks, and harms? How is the science behind this? Can I trust it? Can I get my questions answered? How does my other group, the people that I trust outside of science, feel about this? Does it align with my values as I make decisions around my health or how I treat the environment, et cetera? And do I get a say in how this goes, or is this something that does not value my input? So I'd like to also maybe share some thoughts, again, probably most as a combination of a physician, a community member, or parent, et cetera, about trust, perception, and harm. And I tend to draw heavily over the many years on the work of Stephen Covey in his 7 Habits books, as well as much more recently, the work of Kurt Gray, who's a social psychologist that I think many of you may know. And he put out a recent book lately that I think provides an intriguing perspective on how humans evolved, how our moral minds evolved, how that contributes to our, if you will, fights even about morality and politics, and how we treat the world and each other, and then how to find common ground. So the first 3 points here are really related to Stephen Covey's work, which is the critical importance of trust to effectiveness. And beyond that, the safety that's essential to trust, because without feeling understood, without feeling you're safe, it's really hard to trust either yourself or others, let alone society. It's also important, the framework of the fact that we see the world as we are. Our perceptions of the world is not exactly the same as reality. In fact, it's not exactly the same as anyone else's. So I think we have to keep that in mind as we have these individual and group and societal conversations about environmental biology and engineering biology and how all of that affects the world. Kurt Gray's work is an interesting piece. There was a piece on it in the New York Times, if you're looking for an abbreviated version of it. But he describes a framework where humans evolved mostly as prey rather than the predators we tend to see ourselves as, apex predators, that we are really vigilant for threats rather than looking out for how to potentially hurt others, and that our instinct is always to protect ourselves and those that we care about, that we formed groups and morals to protect against predators, initially animals and eventually other humans that could pose a danger. And as we talk about things where people have different views, when people see something as wrong, it can be much more helpful to ask ourselves or others, what harm do they see, rather than what's wrong with their perspective. And I think as this kind of conversation goes forward, that'll probably be a central piece to making sure that we can bridge any divides between perspectives on this and the educational and fact sharing that we use with others. He also makes an interesting point that's supported by research about our tendency to overestimate the power of facts and the statistics that we use to try to resolve differences when we identify them, and talking instead about the power of stories, with humans being storytellers over the eons, and more recently, the Age of Enlightenment and beyond, turning more towards science and fact that has dominated. But as we can see in our somewhat polarized societies across the world today, that it appears that stories may have the greatest power when harm is perceived and differences are significant. So with that, I'm going to go ahead and turn to some published data. I personally have not been able to find a great volume of public data on views of engineering biology, but there are a couple of studies I'd like to share during my time that I hope will be helpful. One is from the US and one from the UK. This first set of data looks at the US public awareness regarding knowledge, trust in engineering biology. And this one comes from the Woodrow Wilson's Center for Scholars Synthetic Biology project. It was underwritten by the Hart Research Associates, who had done 5 years of annual public surveys and opinions and a 2013 follow-up. So this ends at about that time. But as you can see from the table, there's relative stability in the awareness and knowledge about synthetic biology during that time, with somewhere between 75 and 89% of the respondents really having little or no knowledge of what this is. And as you'll see later, that has not changed dramatically since that time. So we have a lot of education to do about this, but also opportunities to help shape perspectives and build trust. When in this set of survey data, they asked the question, could we apply some knowledge and see how that affects perceptions? The survey participants were read a script I could share later if we'd like, that describes the risks and benefit of synthetic biology. And I think in a way that most of us would probably resonate as being reasonably fair. And then they asked the respondents to rate the relative benefits versus harms of synthetic biology. And if you look at the data here, you can see the answer seems to be that while a greater percentage of informed participants felt that the benefits outweighed the risks, the perceptions that the risks outweighed the benefits increased even more. And very few ended up at the end of that not having an opinion at all or being unsure. So I think we need to keep that in mind as we think about the more people know, how do we make sure that what they know has the right balance of risk versus benefits. The interesting part about this survey was that the greatest negative swing came among those who were initially of low awareness, women who were over age 50, individuals who had had some college, evangelicals, and those who attended religious services regularly, including weekly. So the top public concerns relating to engineering biology are also worth looking at. They looked particularly at those with informed responses after they had shared the information in their script. And they centered around the concerns about creating harmful things like bioweapons, and that risk increased as people became more informed, but they still felt that the benefits exceeded the risk. More strikingly, the concerns about artificial life being morally wrong was much higher when individuals were informed about what engineering biology is about. And the concerns were lower when it looked at issues of environmental damage, and that the informed view seemed to lean towards the benefits being greater than the risks overall. There were other couple of questions that they looked at in this study about other synthetic biology applications that seemed to generate more concern than optimism. And they tended to center around food or agriculture. One, the genetically altered bacteria that release a growth hormone to speed up root growth, and also creating food additives produced synthetically by bacteria. So that, I think, is just an interesting consideration, and I think we'll see more data that talk a little bit about that particular aspect of engineering biology. When asked which one group did the public trust the most to ensure the greatest benefit and the least harm from engineering biology, the public selected university scientists and researchers most often, and the federal government least often. That may not be a big concern or surprise to people these days. I think it may be somewhat of a concern that the federal government is trusted somewhat less. I'm going to now turn to a second study. This is the international study I was referring to. It's much more recent. It was done last year by the UK Department for Science, Innovation and Technology, and was, again, a public awareness, knowledge, and trust survey. It looked at 3,000 UK adults. They weighted it to try to be representative of the entire country, and they looked at 5 areas: healthcare, chemicals and materials, low carbon fuels, agriculture and food, or recycling natural waste. And again, if you look at the combination of people who had never heard of engineering biology or who had heard of it but couldn't explain it, it again ends up being around that 85% that it was back in 2010 and 2013. And only 11% could venture at least a partial explanation. So we still have lots of education to do about this. The interesting thing was across these 5 areas, engineering biology was seen as broadly useful, you can see towards the bottom, across all of those disciplines. And as we think about how we might apply engineering biology to benefit societies, I thought it was interesting that they also asked the question of the same number of respondents, which of the issues facing the country would they say are the most important? And I used arrows to point out that there were several that we could also imagine in our own country here, where if we can use engineering biology across these domains to help with things like the cost of living, health, the economy, or climate change in the environment, we will also probably help resonate with positive perceptions as well as trust in the systems, as long as they meet some other criteria you're about to see. So this table I thought was interesting because it asks the question, which of the following is the most important to you? And what came out on top was that the engineering biology that's deployed would be safe for consumption and use. Probably not a big surprise if you think again about that Kirk Gray theory on the harm-based minds of humans. But it's also, I thought, interesting to see how much how many fewer respondents thought that positive statements from experts or even other people that they trust would matter in their decisions. So safety first, opinions matter some, but much less than maybe many of us thought. On the other hand, overall, again, in the end of 2024, the good news, I think, for many of us is that society generally has a very positive view of this, and that gives us a good place to start with as we move forward with developments and communication about progress, and informing people about risks and benefits. I included this slide. It's a bit of an eye chart, so I apologize for that, but hopefully it'll be useful for your background information. This really provides sort of two main points about specific public views regarding engineering biology. One is almost all of these on the left relate to the public wanting to know more about engineering biology products, and that knowing more will help them make additional decisions about use and favorability, etc. The other is a pretty clear statement that the public also wants to have a say over what engineering biology applications are or are not used for. And I think, again, the issue of trust and progress will depend on societal and public buy-in. On the other hand, I found another article that I thought was intriguing about how engineering biology experts themselves view lay perspectives in shaping engineering biology science and regulations. So this study by Wirtz and colleagues looked at and surveyed 790 US corresponding authors of synthetic biology papers in 2010 to 2015 to assess how their own perceptions of synthetic biology risks and benefits relate to their views of public input, whether the public should defer to scientific authority, and whether regulations would be adequate as opposed to some of these other methods of getting input and making decisions. The results showed that experts who saw less risk and favored greater deference preferred a more closed system where regulations suffice, citizens should not be involved, and scientists know best. On the other hand, experts who saw more risk to engineering biology applications and valued the public's perspective in conversations about decisions favored a more open and inclusive system. And while this may not be entirely surprising, I think it raises an important question and point of emphasis that we should be considering with regard to scientific community bias, which of course all of us have our biases, but also to help make sure that we can get, if you will, on the same page with regard to a balance between the progress we need and the input from the public that I think we probably must have. So we seem to be at a place where right now we're at relatively low public awareness and knowledge, but most people have opinions anyway, and especially if you provide them with additional information on risks, benefits, and what engineering biology is. The public does want to know more about this, but in so doing, we could increase concerns about harms more than benefits, and that this has implications for communication and education efforts as we inform the public and we have public discussions. Public is not swayed much by positive statements from experts or people they trust in comparison to their views on safety and the information they may get from that. Experts themselves have different views of risks and their valuing of citizen input or system openness. The universities and researchers appear to be trusted most. And although I haven't put in many individuals' examples of this, in their survey responses and in some of the focus group open-ended comments, there was considerable discussion about environmental, physical, and moral harm, especially with engineered life, where the concern I sort of think of as a little bit like Jurassic Park, the notion of escape from where it was supposed to be contained, and mutation or change in a way that we can neither predict nor control. And so again, perhaps some of the other speakers here will talk a little bit about this, and I imagine you've had a number of sessions where that's been discussed. Creating life, or as they would put it, playing God, was a significant concern among a substantial number of respondents. And the dual use or weaponization, weaponizing was a significant concern. And that synthetic cells and organisms were examples that were actually specifically described in the surveys and focus groups. So I'll close here by emphasizing what I think this means for public trust and awareness, and that the public is particularly concerned about unknown consequences of environmental release, that we still need to, I think, debate and decide how much we should be using facts, evidence, and stories, and when, as we educate the public about this. The public favors controls to ensure safety, to lower risk, and to address unknowns, and that the public favors transparency, public input, and regulation as needed, and that their overall sense about synthetic cells is they do need to be safe, to have a benefit, effectiveness, to be predictable, controllable, and even remediable if they have unintended consequences, part of the unknown issue, and that they be resistant to weaponization. So I hope these thoughts were helpful. Thanks again for letting me share them, and I look forward to your questions and comments. Thank you.
Okay, thank you very much. Our panel will proceed through all three speakers first, and then we'll have time for group Q&A with everyone. So thank you, Mike. If you can stop your screen share, and I will turn the time over to Professor Schäufele.
Thank you so much, Chris, and of course to the panel for having me while I'm sharing, hopefully, my screen. So hopefully this is now in front of you. And thank you to Mike for a perfect setup in many ways. And I appreciate the shout out to our expert survey data, the Worse et al. Paper. Glad it's useful. What I want to spend my time on is picking up, and really I was quite serious on the perfect setup on some of the themes that we just heard about. One, talking about synthetic biology, about a probably pretty good placeholder and an illustration of what I think a lot of new science looks like, even in the broader CRISPR space, human brain organoids, and so on. And then also pick up on a theme that Mike already queued up, and that is that for a lot of us, and I mean both the scientific community as well as various publics, our preferences when it comes to moving forward some of those technologies is usually an amalgam of yes, the science, absolutely, but also our value systems that govern how we look at those facts. And then I want to end on maybe some pathways on what I think really are pretty good rules, similar to what Mike did in his talk. So let me start with what many of you have heard 1,000 times, the idea of wicked problems. It goes back to urban planning. This is Los Angeles on Thanksgiving. Obviously not an ideal way of governing traffic, right? There are tons of solutions. Moving forward, we could make driving cars expensive, like Singapore does. We could invest in a dense public transportation and subway system in an earthquake-prone area. We could do tons of things, all of which are not perfect. So that's typically what wicked problems are like. There's not a single perfect solution that science can tell us about. Number two, they do require value trade-offs. My right to drive as big a car as I want versus collective outcomes that are less than ideal. And number three, they require normally some form of regulatory policy intervention. And of course, that's exactly what we're talking about here, if that is synthetic cells, or if that is some of the more recent work that has made headlines, and that's probably for a lot of people a pretty good mental shortcut, albeit imprecise and in a slightly different space. And then this just ended up in my inbox, of course, this morning. And as somebody who sits in a College of Agriculture and Life Sciences, production ag and animal agriculture and use of germline editing in that space is one that's probably a perfect illustration of that wickedness, that new type of science where we don't have a single perfect way moving forward, where tons of values collide and where ultimately regulatory or policy action will be necessary in some way, shape, or form, and public buy-in, as Mike said it so perfectly, being key to that. So I just want to show you some data really quickly. This is older data, but with a similar team and some of the personnel overlap to the expert survey that you just saw. This is public opinion data asking people at the time about nuclear energy as kind of a legacy technology, and then nanotechnology, which raised some of the same questions. Can we create materials that don't exist in nature, for instance? Chris knows the space well. And then the synthetic biology area. And we asked tons of questions, but I'm just showing you breakdowns on two of them here. Do they conflict, either one of these technologies, with with religious views? So phrased moral and religious for folks who are not religious. And do they blur lines between humankind and God? And you can see how, as we go from legacy media into synthetic biology, that these concerns increase, and that values really become more and more of a centerpiece. I'm not going to talk a lot about motivated reasoning today, but many of you have heard about confirmation bias and other things that illustrate really nicely how these values end up shaping how we interpret even scientific facts very, very differently. That's also why, and I mean that quite seriously, a lot of the work that we have done as scientists when it comes to communicating, and I'm singling out people here, but I don't mean that as criticism of their work, but just as illustrations of things that tend to not go particularly well. There was a great article in Gizmodo about Bill Nye the Science Guy and his show on climate, where he basically told people, get your together, your proverbial one. This is a real crisis, but most of us don't respond particularly well to being yelled at by scientists, or, and this is Neil deGrasse Tyson, the date on this tweet is really important. This was a tweet at the time before X came along. December 25th, on this day a long time ago, a child was born who by the age of 30 would transform the world. Happy birthday, Isaac Newton. Now, I think that's pretty funny, but I also have zero religious infrastructure in my cognitive system. But trolling Christians on Christmas Day and then turning around and saying, hey, by the way, listen to us on these value-based discussions when it comes to synbio, to stem cell or other things, is a difficult conversation opener, as is Michael Mann, climate scientist now at University of Pennsylvania, who routinely tweets that this is a fight between good and evil typically the Republicans, and nobody who is Republican is safe to have in Congress. Again, that is a conversation opener to now starting a conversation with a public, and that is a more conservative part of the public about climate change, probably not going to be the most fruitful path forward. And of course, the same thing for Richard Dawkins, who routinely mocks people who are either religious, and again, about 50% in the US of the population self-identifies as religion being very important in their life. Probably not a particularly good conversation starter. It's actually our kryptonite. It's what draws a red line between where we are and where we've seen, and where our value system is seen, and where the public would like our value, or different publics would like our value systems to sit. So that's, I think, ultimately a problem that has created, and some of you have seen these data. These are out of the University of Chicago trend data that have gone into the old chapter 7 of the science indicators. And so over time, science has enjoyed unbelievably high levels of trust, second just to the military. And the military just pulled ahead of us around 9/11. So we've always been at the very top of the most trusted institutions. I want to highlight two things here. One is right after COVID, trust in science. And very often we blame that on declining levels of trust. And there's some truth to that. I'll get to that in a second. But the gap here that we're seeing between the blue folks, and those are people who self-identify as big D Democrats, and the red folks who self-identify as Republicans, and the gray folks who are independents, which in the last election were close to 50%. So what we end up having is we have a widening gap that partly is driven by the gray and the red lines that are going down. So these are independents and Republicans making up about 70% of the population. So 7 in 10 Americans are sitting at that red-gray spot. And then for the blue line, what we're seeing now is this is basically Democrat, big D Democrat voters saying, Trump is anti-science, so we're going to be pro-science. We're going to show historically high levels of trust in science because it becomes a political messaging tool for us. It becomes part of our political or partisan identity. And the moment we end up at this spot where we see partisan gaps, where Democrats claim this as part of their identity, and Biden and Obama both, when they came into the White House, said science is back, signals to the rest of the country, and those are both independents and Republicans, that it's not for them, number one. Number two, and this is maybe more important, since then, we've seen really independents have recovered a little bit, but we've seen declines in trust, and the recent Pew data show the exact same thing among both Republicans and Democrats. So we're seeing declining levels of trust. Why? Because we're now becoming just one of many institutions. Science claims that it's a unique institution. We curate, create, and share knowledge in ways that is neutral, that is objective, that is free from partisan biases. The moment one party co-opts science as being part of their identity and the other one sees it as not part of their identity anymore, we're going to end up with exactly the kind of partisan dynamics that we're dealing with right now, and the undercurrents of which I think will last well beyond the next 4 years. So that's all the depressing stuff. Let me hopefully shift into a bit more optimistic stuff, and that is, I would actually make the argument that as we're thinking about where trust needs to be, very often we're operating almost from an assumption that we want to have 100% of the public trusting science. I would make an argument that that's not what we want. And this is a poorly drawn, hand-drawn graph. I fully own that. At the bottom, so if the y-axis is a healthy science society interface, right, where we're making good decisions, science-informed policy decisions. If none of us trust science at the very left, we're in a bad spot. It damages society. I would argue if all of us blindly trust science, it's the exact same situation where, as we're pushing forward human brain organoids, as we're pushing forward applications of CRISPR, of AI, that will fundamentally transform what it means to be human and how we live together as a society, we do need some of those debates, and Mike already talked about the public wanting that buy-in. So the point being, somewhere in the probably upper end of that curve, there's a sweet spot where we're maximizing democratic benefits, having a healthy level of skepticism, but also a deeply, deeply rooted trust in science as a tool and method, as the best way of creating, curating, and sharing knowledge. Very often, the second, I think, fallacy that we're getting into as we're talking about rebuilding trust is, and this was certainly post-COVID, I think this is also certainly in the biomedical field, which much of this work relates to, we refer to historical missteps, and this is a euphemism that doesn't do justice to what happened with Henrietta Lacks and with the Tuskegee experiments, and so on, and we all know those well. But that's really only half, or maybe not even half, of the story. If I'm an African-American mother in 2025 in the US, I have a 3 times higher chance of dying from childbirth, either pre or post-birth, than a white mother. If I'm rich, if I'm wealthy, I have a 2 times higher chance. So me not trusting the medical establishment is actually a pretty good risk calculus. And rebuilding trust is not, that's just dealing with a symptom. It's not dealing with the underlying cause, which are inequities in the health system. So a lot of the rebuilding of trust is really at the, and I would argue very strongly also as we're pushing forward certain applications in bio and related spaces, is really about making sure that we do that equitably, and avoid the very missteps, and again, I'm phrasing that euphemistically, that have led to that lack of trust, rightfully so, I would argue, in the first place. And that typically leaves us with the last one: we need to listen, right? And I think everybody nods typically, yes, absolutely, we need to listen more to communities. But we haven't quite figured out what that looks like. This is a piece that we put out in PNAS a little while ago, where we basically outlined the bandwidth of what engaging with different publics can look like and has looked like. And very often, of course, we end up on this left kind of side. We need to avoid public controversy. We don't want to go the ways of GMOs. That's typically the scenario that people conjure up that was horrible and it didn't work. And I would agree with most of that diagnosis, frankly. My early career at Cornell, I spent most of my time on plant GMOs and public perceptions. But if you go toward the right on this graph, you get into educating the public, and it's also pretty important, but it's still very top down, right? Maybe have some deliberation and bring people in and they can talk about it, increase maybe even representation. But at the very right is actually a scenario when we listen and the public or different publics tell us that they're really deeply uncomfortable with an area. Are we willing to actually listen to their objections? When we did the consensus study on the future of human genome editing, this is a long time ago now, the hard of hearing community came to us and said, look, glad that you want to fix us, and that's exactly the word they used, but there's nothing wrong with us. There's nothing that needs your medical intervention, and there's nothing that needs your germline or somatic edit to help us with what you call as a problem and what we see as a core part of our identity. So are we actually willing to listen to the point where we let public input govern the way we do science or the kind of applications that we put forward? And I leave that as an open question, but it's one that when we commit to listening to communities, I think we're often leaving out. A second one is that we often talk about finding trusted messengers, and I think that's a core part, right? Messengers in communities that can help us connect with communities that are traditionally underserved by science communication and outreach and education and engagement, excuse me. And that's a piece we just came out with. I just want to show you the data because they're really deeply depressing but also show us where work needs to be done. So what we ended up doing is we ended up basically hiring actors who were introducing CRISPR as a new technology, especially for curing particular diseases. We worked with filmmakers. It was a pretty cool project. And they all had the same name, were described as scientists at Wisconsin, except for they introduced two different applications. One was Ruthie on the right, a patient, Ruthie, with albinism, and then on the left, a patient with sickle cell disease. And last name was Sanchez. I'm totally blanking on his first name right now. And what we wanted to know is, of course, who are these messengers as scientists try to connect with different groups. And we basically manipulated gender, so male, female, in this case, somewhat narrowly, male, female, and then African American, Black, and white scientists. And here are the breakdowns, and I'll just show you two numbers. And what we wanted to know is in terms of trust, how much warmth and expertise. Many of you know Susan Fisk's work at Princeton on that. And so both how much expertise did people see and how warm did they see the scientists were. And I want to highlight two findings here. One is this is a Black female scientist treating a Black child. So all of these are treating the Black patient, and you already see that Black females are rated pretty low, both in terms of warmth and competence and expertise, excuse me. But at the very bottom is where Black female scientists introducing the clip for a white patient. So when you shift over from a Black patient to a white patient, that's when things really bottom out for Black female scientists. Again, everything is absolutely constant. We had multiple Black male, Black female, white female scientists to average out personality difference or attractiveness or whatever else could confound these findings. And it holds. It's one of the most deeply depressing findings about how much intersectionality there still is for the kinds of messengers that we need to really go into communities and get some of these messages across. And the continued disadvantages that some of the messengers that we need the most are facing are really troubling, and I think something that we need to tackle. So last thing that I'll end on, and that is language, and some of you have heard me talk about this before, but it's also a way out of a lot of the things that we're talking about. Mike already queued up the idea of storytelling. Right, this is in Frankenfood. This is an article in the Atlantic, actually pretty optimistic about the future of genetically modified crops for addressing world hunger and others. But you can already see the Frankenfood and the carrot, the stitched together carrot. Why is that so powerful? Because simply by adding the word Franken, I tell that story that Mike talked about earlier. I immediately connected to a mental image, a mental frame or schema that we all have. It says scientists are going too far, getting out of control, it's getting out of the lab, and it's unbelievably hard to rein back in. And of course, for SynBio, this is exactly what came back, right? Frankenstein in the test tube when J. Craig Venter claimed to have created life in the lab with synthetic DNA inserted into a live bacterial cell. And sometimes, and again, this is not criticism, it just highlights kind of how complicated or how difficult this sometimes is. If you look at Sternberg's and Doudna's book, Cracks in Creation is an unbelievably capturing or kind of a title that captures imagination immediately, but it also, of course, puts at odds our breakthroughs in science with belief systems around creation and religion and with very little, I think, appreciation among some of us at least. I know that Jennifer has thought about this quite a bit, but among some of us at least about what that may do long term. But framing is also the one that helps us make those connections. And this is, I promised the last slide, that helps us make these connections on explaining why these technologies are actually important, why we should pay attention to them if different publics, why should we start worrying or paying attention in an excited way to synthetic biology. Why are they worth the investment, the long-term investment? What is the promise of and Michael J. Fox and Muhammad Ali and Parkinson's, of course, being a great story, but a way of framing it around long-term cures and so on and so forth is something that people can meaningfully connect to. We're seeing some of those frames being used again now for discussions around NIH funding. And this is maybe the most important part, even if we don't share the values that some publics bring to the table, framing messages around the values that are important to them rather than the values that are important to us as scientists is crucial. And I think for COVID, it was a great example where we framed a lot of our messages around loss and around the potential loss of lives and other things. But people really cared about COVID, and many people cared about COVID largely as an economic issue. They're losing their barbershop and other things. And so framing it around those things and saying, every time one American doesn't wear a mask for a day costs us $54 in GDP, is for those people a much better way of presenting it. And I think that's a huge opportunity also as we're thinking through issues that are clearly at odds with some sets of public values. And so framing them toward those rather than as at odds with them will be crucially important. So I'll leave it with that. Hopefully I didn't go too far over. Appreciate the opportunity and looking forward to the discussion, of course. And I'm going to unshare.
Okay, thank you very much. With our time remaining, I do want to give Christina as much time as possible before we get to Q&A. So I will turn it over to her.
Thank you so much. Thanks, everybody. I'm going to take Mike's point very seriously about how facts don't change opinions. I'm not going to show you any data at all. I'm going to talk more about my lived experience as a synthetic biologist over the past 20 years. The majority of that time, not only as a scientist, a PhD synthetic biologist, but also as a science communicator and a marketer, communicating the science and the business of synthetic biology. As people said, I spent 10 years at ECO, starting there when the company was still pretty academic and 20 people, and helped grow that company to be a publicly traded company with hundreds of customers across food and ag, health and medicine, and the consumer spaces. Today, I run a small consultancy on sort of strategic communication for emerging tech founders, primarily in the AI for science space. So this question about perception, trust, emerging technologies, and the stories that we tell has been really, really, really important to me and to my career and to the field that I've been part of. It's I want to use maybe my few minutes that I have here to maybe reject the premise a little bit and continue, I think, a little bit of the story that Mike and Beecher were saying, and in particular highlight some of the traps that we often fall into as scientists when we think about these questions about public trust and public trust perception, and how we navigate both around these stories and these products of new technologies and how they reach their audiences. I read the paper that you all, I think, were reading about synthetic cells from Dr. Rothschild. It's a really interesting paper about this new kind of emerging technology and the potential of the field. The paper ends with this sort of discussion about like non-technical challenges that we're talking about, including outreach and education. I'm picking on that paper because it's a very, very, very typical kind of characteristic way that scientists, in particular synthetic biologists, think about the way that we communicate why education really matters. I think it sort of, again, echoes what folks have been saying. Engaging the public in research and technology development is crucial. Emphasizing education with both policymakers and the public will not only support effective policy frameworks, but also enhance the acceptance and impact of new technologies. Okay, so I'm gonna like unpack that a little bit and sort of like, I think, illustrate what is, again, like the typical mental model of scientists when we think about public perception and trust. Like there is an emerging lab technology, like inside of the lab. For that to go from the lab to impact, you have to sort of pass through this gauntlet of public perception and public acceptance. And so it's a very linear model, and a very linear model that also can be addressed, can be solved, can be like passed through with like the right education. Like if we were just good enough at like framing the knowledge exactly the right way, if we presented it in the right way, if we educated people the right way, if we filled their minds with the knowledge that they needed, they too would accept this technology as much as we do. So it turns out that that's like completely not at all how people make decisions and how things happen, how technologies move from the lab to the market. And so I know that is how we like wish it did, that it would be like, oh, it's so easy. If I just could just say the right words, like we could get there. But there are no right words. Like the only way out is through and through like making things that really matter to people, that solve real problems, that can be like, that are acceptable and that are like trustworthy by being trustworthy, like being vulnerable, being accountable, like showing up for it and sort of demonstrating value to people, value and values. And so instead of that straight line, I'll give you like another kind of framework that I'm sure many of you have seen. You've probably seen that Gartner hype cycle. Okay. The whiteboard. Okay. You guys see that? So like on the y-axis, you have expectations, right? So like, and then on the x-axis, you have time. Right? Okay. And so early on, like there's no product, there's no technology, there's only stories of what the technology could be. And we have a lot of expectations. And those expectations are both positive and negative, right? It is the time where we discuss the promise and peril. Like in both cases of promise and peril, the technology works. Right? Like it works, it succeeds, it scales, it's like doing its thing. And either it saves everybody and solves every problem on Earth, like, or it kills everybody on Earth, right? But this is like the domain of stories. Okay. I'm gonna put see where I was going with this. All right. So and so like because of that sort of like focus on like perils that comes up, the way that dynamic sort of happens like at that peak of inflated expectations, we often again like sort of like, oh no, it's safe. Like let me show you the facts about how it's safe. Because again, we have that sense that if we can fill that gap in people's knowledge about the actual facts of the safety, then we'll get on with it and we can like pass through the inevitable trough of disillusionment. I really liked, I think Stephen called it the chasm of relevance. I thought that was really interesting. Or there's also like the valley of death in technology development and companies that we build. Like this is also very real because once the stories kind of fade from our imagination, once there's a new technology to take its place, to be that sort of like that promise in peril. Oh, yeah, I have another thing to say about that. We start, the real work begins. Like, how do you actually make a useful technology? How do you actually understand like a niche and a problem that needs to be solved? And how do you like actually make it and get it to market and get someone to pay you money for it? Like, those are really, really, really hard things to do. And they go well beyond sort of stories and things about the facts of the safety of the technology. I'll get to that later. But I wanted to say, like, I think I don't need to go into it in very much detail because I think Mike and Dietrich did such a good job of talking about this question of values. Like in this time of storytelling, it's again, like not at all a question of the technology itself that we're arguing. It's in fact like a proxy battle about values. So questions about what counts as progress? What is the role of humans in nature? How should problems be solved? What kind of future is desirable? Who gets to decide what kind of future we all live in? Who gets to decide what is or is not a problem to be even solved? Like the hearing loss case or the folks who have hearing loss, right? Like what counts as a problem? Like, and who gets to decide? That's a values judgment, and that's a question about ideology. It's not technology. And so as we sort of like, again, as those ideological battles like move on to a different kind of technology, now maybe it's AI, maybe it's something else, like maybe it's synthetic cells. I don't know, people are trying to make that happen. Like you also have to like make real products in the real world. And so like I've lived all of this, like in synthetic biology, we all like for a long time. And so like we need to kind of go from this time where there's nothing tangible to actually accept in public acceptance frame, except for ideology, into something that people can now actually choose to spend money on. And so like I want to tell a little story about the mascara that I bought yesterday as one example of this happening in the real world, a real example of this. 15 years ago, like, like late noughts, 2010, Lady Gaga is singing about bad romance. Like folks in sort of the academic environment of synthetic biology are saying, we're going to revolutionize every material and chemical in the world. Like, we're going to completely change the entire industrial manufacturing base. We're going to refactor everything. We're going to make everything sustainable. We're going to solve every problem. It's going to be awesome. I believe them. I'm doing it. Like, that's my job. I was like, hell yeah, we're going to do that. We're going to do synthetic biology. Those folks started a company called Bolt Threads. They said, we're going to make like Kevlar. We're going to make these incredible materials from spider silk. We're going to like, again, totally reframe the entire like textile manufacturing landscape of the United States and beyond. It's really hard. Those things don't change readily. Like, those are markets that are very entrenched, that are hard to change, hard to move, like they have to reach a very, very significant scale. But those proteins are really interesting, the ones that come from silk. They're really impactful. They can be used for a lot of different kinds of things and properties. They make really excellent mascara. And so now Lady Gaga herself this is from House Labs, her brand, saying like, this is biotech enhanced mascara. That's in the like description on the website. You can buy it for $29 plus shipping. And she's on TikTok talking about how plump it makes her eyelashes look and singing songs about lashes. And so I'm belaboring the point to just say like that these technologies that like cause this like very, very like big ideological story about like what the future could look like end up being quite mundane and everyday experiences. And those questions about how does somebody choose to buy biotech enhanced mascara? Like, I think they have to sort of assume and trust that somebody down the chain is going to be held accountable for it being safe. They trust that Lady Gaga is not going to put her name on something that's not safe. They're gonna trust that Sephora isn't going to do it because they're going to get sued if they don't do it or something, right? And then they make a choice based on like a lot of other things. How does it make me feel? Can I afford it? Is it good? Is it actually better? Did my mascara run out? Like, did I like that TikTok that she made? And so that's sort of like real world of like the choices and the technologies, like, and our actual lived experience interacting with these products. I have many more that you can play with. Like, I think is going to look a lot simpler. So this sort of like lower hype level, like the plateau of productivity, that looks like infrastructure. It looks like things that are invisible. We don't call nanotech anymore. Now they're just LEDs and like our TV screens and things. There are sunscreens. Like those are real technologies that have had a real impact, have built great companies and made transformative impacts in the way that we do everyday things. But we don't have to talk about them anymore in that sort of proxy and ideology. But we should also sort of understand, again, like what people want and need, how those products move and how those stories move and translate. And so I think that's all I wanted to really say. So thank you for your attention, and I look forward to a very rich and complex discussion.
First of all, thank you to all three of our speakers. I think it's been an excellent panel. I know that we are going to be a bit stressed for time, and I will defer to Emily and Tricia there. But I do want to open up for questions with the committee, and I also know that Emily and Teresa are going to help me with questions from those in the room, since I too am a virtual remote participant today. So anyone in the room have a question?
So many questions. I think we just all, this was a wonderful session. So thank you to our three speakers. You've just done a fantastic job. So if I missed, I have so many questions, but I'm going to limit it to one in the interest of time. I'm Felicia Wu from Michigan State University. Deidre, I was very much intrigued by the graph that you had showing that it's certainly not good that none of the public trusts science at all. It's also negative if 100% of the public trusts scientists. Now, in the role as an economist, we oftentimes try to optimize some sort of level, whether it's the price of a product or the amount that is consumed. Has anyone ever attempted to make any sort of estimation as to what proportion of the public ought to trust science for a successful and thriving society, whether it's a nation, a city, etc.?
Yeah, I'm not aware. This is a really good question. Thank you, Felicia. I'm not aware of anybody having done this. I think the challenge will be for whoever ends up doing this is that there are two variables that are going to differ depending on or that will make that relative optimum different at different for different types of technologies, right? Where some of them where the science is settled pretty quickly early on, and where some of the debates are really in either the value space or some of the application spaces and equitable access and so on. But then I think that relative optimum is also going to differ, or maximum is going to differ at which point of the issue cycle. And folks at Arizona State, David Gustin and others during when Nano first came along have talked a lot about public engagement and kind of a lot of that skepticism being really important early on, as we're in a stage of what they call upstream modulation. So where we kind of can still modify how the technology potentially develops with public involvement in that decision making. And then later on, more downstream, it's probably, hopefully, there's a much more robust level of trust in what we ended up with as a result of that skepticism being built into the process early on. So I think that relative optimum is sitting at different times in the product, not in the product life cycle, in the issue cycle of the technology that we're talking about. And then, as I said, I think it also differs across different types of technologies, if that's vaccines or if that's anything else. But I'm not aware of anybody actually having tried to quantify it.
Yeah, and I would add, this is David Dunning from the University of Michigan. I would add there are other complications that really matter. I mean, let's say it's 50-50. 50-50 matters if you're talking just about a mix of people. Or if you're talking about 50% of Republicans over here say one thing, 50% of Democrats over here say another thing. Now you're in a completely different set of circumstances. So there are a number of variables that go into what is the optimum.
Fully agreed.
I see that Jason Delborn has a question. Jason, please.
Thanks. Yeah, I'm from University of Wisconsin, fellow Badger with Teach Room Now. Question for Christina. So the hype cycle that you described, it seems pretty toxic and unhelpful. So the question is, maybe broadly, how can the scientific community act differently to not try to get to that peak of hype, which doesn't seem like very productive conversation? Is there a way to avoid it? And actually specifically for a committee like this, how do we not contribute to that if we are addressing a technology that is early in that time scale? Can we lessen the peak? Is the fall not as painful? Do we have any examples where we don't see this pattern around an emerging technology that does have promise to do important things, like better mascara?
I can give my perspective. So like I have been accused by many scientists of committing the crime of hype. So like take it with a grain of salt. There is an amount of like energy and promise and like bringing people to you with the same shared values, with a shared kind of vision, bringing the investment that you need to like build new technologies, where you do have to tell that story of I have found in my experience, at least to like avoid that sort of like very explosive, like Frankenfood, like playing God sort of story, is to sort of really, I'm maybe really on what Teacher said, like pull on the things that matter. Like, okay, I'm going to talk about how we're going to invest money and build a joint venture to build strains of bacteria that can fix nitrogen. Like that's a real project. We really wanted to do it. Like there is a real problem with how much energy and greenhouse gas comes from nitrogen fixation. Like it's worth investing in that kind of technology. Here's why it matters to us. Here's what we're doing. Here's the kinds of, like it's going to take a long time. It's a moonshot, and sort of like articulate those stories. Like those navigated, I think, pretty well when in the media, like we had a sort of like good conversation about like, oh, what's the timeline? What's the risks? Like, what are you thinking? Well, we're investing like $100 million into this project. We're partnering with Bayer. We think that there's a real promise, but like it's risky technology. It's like new technology. And so like that question of like, oh, is it safe? Like, should we do it? Like didn't even come up because we really centered it around the values. Like we think this is more sustainable. We want to think about the application. The other piece of it has been like, I made these stickers and I give them out to everybody. Like I have a few left over if you go and want some, like saying like, I heart GMO. So again, this isn't to like rehash the old debates, but to sort of say like, here's what I think. This is why I'm investing my life energy into doing this. Like, here's why we're doing this. These are what I value. This is what I think we can make GMOs that you'll love. And like, we want to talk about them. I think another part of the way that it became toxic too was like, oh, don't talk to me, or like it's fine, it's safe, trust me. Why don't you trust the science? And so like that became, so it may still be hype, but it's a less toxic kind of maybe. Yeah.
Okay, I see Robin has a question online.
Yes, thank you. This is both, I guess, a question and invitation for reflection. We sort of, the framing idea of this panel has sort of been the power of stories and possibly the way that those stories run in tension with what experts sort of think to be true based on data. But what are the stories that scientists are themselves telling? Like I sort of this cleavage of like scientists are using information and the public uses stories. I think that stories are very powerful. I think there's many of them present in the scientific community. I think this is probably important to reflect on in terms of questions of trust, given that we've been told by earlier panels that the people best able to anticipate the possible risks of new biological technology are themselves scientists. So I just sort of wanted to push a bit on that. I think it sort of builds out of how to think about the hype cycle as a way of, informing our decision recommendations we might make, but sort of a bit more how that filters into thinking about how scientists are also framing these questions in their discussions with the.
Yeah, maybe I could comment about that. As I look at the hype cycle and I think about the comment, the part that intrigues me and is a story metaphor, if you will, for it is each of us in every romantic relationship we have ever had probably goes through a hype cycle, right? We see the potential, we really magnify it. We actually may try to encourage investment in the relationship, or in this case, engineering biology. Then good news, we have some, and the voices of quiet concern are quiet. We do have our worries, but we don't articulate them as much as the hype part does. And those investors who get on board understanding the risk will still nevertheless often get on board. But the price we do pay for it is the issue of disillusionment. I've implemented enough electronic medical records to see that, right? We really needed to do better. Oh my gosh, we have it. Now we got to fix it, right? And so I think the only one that really lasts in the end, I think to Christina's points, those are the success stories, the ones that reach that productivity plateau, and we now start to work with them without having to think about them. So I think we want to do that. The story part, I think for the scientific community, is we have to be careful. As objective as we try to be, none of us is objective, right? So we all want our projects to work. Yes, we want to seek the truth. We just hope that what we're working on turns out to be the truth, as objective as we try to be. And that's part of why I use that words paper, in part is to recognize that we are also humans, and we have to be self-aware about our own role in, do we know best? Does the public know best? And what do we do about the tension that exists between those two? So Rob and I really appreciate your perspective on that, and I do think we need to address it.
Okay, thank you. Oh, go ahead.
It's okay. I think that your question is really important, Robin, about like the questions that the stories that the scientists tell themselves. I know, like for me, like the story that was very, very dominant in synthetic biology and sort of remains like, again, like a driver for a lot of people has been like, we're doing real engineering finally. Like we're gonna finally be able to like really control biology, and like therefore we will be able to do all of these amazing things. And I think that story like motivates a lot of people, but it is a story. I think it has an ideology attached to it, and it may or may not be actually factually accurate about how we can interact with biology. And so I am very interested in those stories that we tell ourselves also, and like I think it's important for us to sort of also unpack those and ask like, well, yeah. I think the synthetic cell version of that like is like trying to push it even further. So for folks who are thinking about synthetic cells, it's like, oh yeah. It actually turned out it was too hard for us to fully control biology by just saying we were going to do it with BioBricks. And so like, what if we just got rid of all of that stuff in biology, and like then it will be easy and we'll finally be able to control things and make things look the way we want them. And so I think that's another part of the sort of hype cycle that like down here is like when you're like, oh, like things don't work exactly to plan. Like things have to like adjust the plan. Yeah, the medical records, maybe people don't like filling it in. Like it does, like there's bits and pieces that aren't working, right? Like that you have to sort of be constantly changing your plan. So yeah, what we do in the lab like will not inevitably go in a straight line to the product. And so I think a lot of the story of new technology is like to try to reinforce that, but it may not be true. So like, yeah, that may be another thing to think about. Like, what does it look like to be continuously like involved in how things are moving into the world? How does it look to sort of take that feedback from the way that the product is working, from how it's filling a need, from how it's serving people? That also fits the story of Frankenstein, which maybe I'm not going to tell you a lot more about another time.
Okay, I think we have time for one last question. So Pete, I'll turn to you, and the floor is yours for a final question.
Thank you. Pete Carr from RTX DVM Technologies. My question was for Christina. I was really struck by what you said. There are no right words that really hit home. As somebody who obsesses over words sometimes in particular too. Not that there isn't a point to that, but
my job is words,
yeah.
So I get it. Yeah.
But and your point with that of the path to being trusted is by being trustworthy. I was wondering if you could elaborate a little bit on what the pathway is to being sort of demonstrably trustworthy. I'm not asking you to say, can you teach me how to fake being trustworthy?
Yeah.
But what is that pragmatically, that hard road look like more?
Yeah, maybe I guess from my experience, like I think again, people are obsessed with like there should be a right word. So like, I would literally get phone calls from other marketers being like, stop saying synthetic biology. Can you please? We have decided to call it precision fermentation now because we think that that's going to be less scary and that's going to make it work. Didn't make it work. Right. And so it doesn't matter. Like, again, the word doesn't necessarily matter so much. Like, I think it really is about sort of, yeah, showing these other. So I think in practice, what that looks like is, again, like when somebody expresses a concern, like that is the perfect moment to like demonstrate like a vulnerability and openness, like a willingness to listen. Like it can happen at like really at that one-on-one level, like where you're not immediately like, no, let me explain the facts to you. Like that's like the fastest way to lose trust or sort of and stop being trustworthy. And then I think you can kind of like extrapolate like, well, what does that look like now as I make my website for my company that's trying to sell these things, as I make my like write my papers or interact with the media. Like it is about a willingness to interact, to ask, to treat that concern with curiosity, like rather than with like a like distance. And to sort of like, again, try to connect with people. And so being vulnerable, expressing your emotions and values, and like being like in the conversation, I think is like the best way it's the only way to build trust.
Thank you.
Mike, please. Thank you.
Yeah, if I could just make a quick, maybe take-home way of putting that. Stephen Covey put this term of trustworthiness as a combination of character and competence. And in my medical world, when I talk to students and residents in that, they want to know, for example, if you're a surgeon, will you do the right thing? Do you have the character to make sure that what you're going to do is the right thing from the perspective of the patient and others? And the other is, are you competent? Will you do the thing right? So the public is counting on us to do the thing right, which is make safe, effective, useful products, and that we have the character in it to make sure that that we are representing their best interests. And Christina and Dietram's comments about this are really spot on for helping us get there.
And Dietram, final thoughts here.
Yeah, just really quickly, I think in the study that I showed on African American white scientists, we used an operationalization of expertise, of competence, of perceived competence that is both warmth and expertise. That's the idea of Susan Fisk's work at Princeton. And the important part is competence is a large part of the story, right? Getting the science right, absolutely. But the idea of warmth, and that is exactly why I had that one slide that says, you opening up by signaling to everybody that your values don't align is the exact opposite of warmth. So the idea of endorsing values and concerns, even though I think they're scientifically not merited or that they don't align with my own values is really key. And saying, I'm willing to go with you on your argument on vaccines and on vaccine schedules being ridiculous. And then we're going to disentangle that together, but I'm not poo-pooing it from the get-go. So that warmth part is probably as important as the expertise part. I just want to add that as kind of the last thought on this. Thank you.
And my final thought is none of this is new. This is all circling around Aristotelian ethos from thousands of years ago with competence, character, and dynamism. These are all the same values that we've been talking about for eons. And as we see throughout this panel, a lot of what we face are legacy issues, and they're ones that certainly go across all kinds of biotechnologies, and certainly will be things that our panel faces and our committee faces going forward with our charge with the National Academy. So another warm and welcome round of applause for our speakers. Thank you all for a wonderful and dynamic panel.
Okay, thank you all. Thanks again. And now we're going to be moving to our closed session lunch. And thank you again to our speakers. Great to see you all.
Thank you. Take care.
Yes.